Good day, everyone. I'm Dr. John Ward. I'm the director of the Coalition for Global Hepatitis Elimination at the Task Force for Global Health in the United States. I'm also affiliated with the National Cancer Institute at the NIH. It's my pleasure to introduce you to day 2 of vaccines against hepatitis C virus, harnessing immunity to eliminate globally hepatitis C. We had a great set of presentations and discussions yesterday that we will be continuing here in a few moments. Vaccine development is a critical technology that's needed to eliminate hepatitis C around the world. It's very challenging to eliminate an infectious disease without a vaccine. We have highly curative therapies for those who are already infected. We now need a vaccine to reduce the high transmission risk for populations such as people who inject drugs with an incident rate of 10 to 15 per 100 person years per year of their injection history. And even the risk of transmission in countries where unsafe injections in healthcare pose a risk for those in healthcare for those countries. And so prime opportunities where vaccine can play a role. We're going to be continuing to explore the technical challenges in that development. For those of you who missed day 1, just to remind you, just to let you know and remind others that all of this information will be available on the Coalition website at globalhep.org in a variety of different forms as shown here. And it's coming together with the other webinars that have been part of the series beginning last year and that will continue this year in the future. So please continue to participate in this webinar series. If you're registered for this one, you will receive notice of the other ones coming up in May and June and the months thereafter. Here's the program for today, and you'll be meeting obviously these speakers, and encourage everyone to remain for the panel discussion, where we'll have a very lively discussion on a variety of different topics that have been touched upon yesterday and today. At this point, I'll turn it over to our two moderators, Chris Walker and Georg Lauer.
Great. Thank you, John, and thanks again to you, Lindsay Hebert, and the rest of the team at the Coalition for Global Hepatitis Elimination for making this possible. I look forward to moderating the session today with my co-moderator and colleague, Georg Lauer from Harvard. Our first speaker of 4 today is Dr. Mansoon Law from the Scripps Research Institute. The title of his talk is Next Generation HCV Vaccine Antigens, What We Know About HCV Neutralizing Antibodies and Responses and How to Leverage Recent Knowledge in Vaccine Design. Mansoon?
Can you hear me?
Yes, thanks, Mansoon. Yes.
Yeah, okay. Yeah, thank you so much for the invitation and provide me the opportunity to share my thoughts about this HCV vaccine problem. So to this audience, I don't need to introduce too much about the cause of HCV infection, and over a long period of time, it will induce a lot of inflammation and reinfections of the liver, changing some architect. And one of the key problem really is the HCV, some known as the silence mutation, silence epidemic, and a lot of infections ongoing, don't know. And having a vaccine pretty much can help stop many of these stages of damaging the liver in the infected patients. So one of the thoughts that my title is talk about the next generation HCV vaccine antigens. So we know that there are 2 major vaccine candidates being tested in humans. One is the viral vector vaccines to stimulate protective T-cell response, talked by Andrea Cross yesterday, and then the E1, E2 candidates to elicit neutralizing antibodies, led by Mike Houghton. And so so far we know the current forms we're not quite sure, but probably not protective, or we haven't have data to show they can be protection. And these two vaccines, I think there's still a possibility to make them to work if we be able to really maximize the immunogenicity and make it elicit very potent immune response. And we did talk about a little bit about the maybe combining these two vaccines or one form or the other, eliciting both T cell and B cell would be a good approach. So my thought is if this vaccine works, it's great. But if not, we really need to think about what to do. And then that's part of the reason I think we need to have a better pipeline for HCV vaccine to increase our chance. And that come to a point that I think by understanding better the immune response being elicited in infections, also being elicited in vaccinations are important and for the design of next generation HCV vaccine. So my talk is pretty dense, but I try to provide a more overview and a high level way to look into the problems. And in this case, I try to cover 4 different topics. So the first one is about like what's the basal immune response required for protection. So far, the immune correlate is not clearly defined, but because there's not a HCV working HCV vaccine, so we just based on the information we know T cells are important, neutralizing immune response important, and what the problem is we need to have better resolution or details about those immune response. And Andrea yesterday talked about the T cell response, very interesting and in detail. And I'm going to talk a bit more about the antibody response in the past studies. I will also cover a bit about the different topics I will go on in my talk. So the Chiron discovered HCV with other investigators, and they make this full-length E1, E2 antigen. So a few years ago, I was able to get some of these antigens from Phil Domenza. At that time, he was leading the Novartis vaccine effort. And we done some analysis of this vaccine candidate. It come out very nice, like as a major peak. It's about 670 kilodalton when you run a size exclusion. And the protein cell is very clean. You have to see this E2 band and E1 band. And it formed this non-covalent E1, E2 complex. And it just separate by cell with just SDS without reduction. And you can take this protein and look under EM, and you can see it got a kind of lipid micelle with this lob sticking out of the E1, E2 complex. And it cells actually very good in terms of antigenicity. You can pretty much put 3, 4, I think we try up to 5 now, but this one you show you can put 3 different antibodies fragment. You see they all bind to it. And it actually preserves the antigenicity of the E1, E2 antigens very well. So the studies we did, it's very lucky with the help of Sharon Fei and Rajan Koshi. We brought back all this vaccine anti-sera from that study in the phase 1 clinical trial. That at that time 4 doses, test different doses. In parallel, when doing our analysis, we went to immunize. In our case, it's relatively small animal size of 4 monkeys with this. In this case, we immunized 5 doses of it, and in between, we also collect some sample to isolate monoclonal antibodies with the help of Robert Lanford. And overall, the immune response quite similar. I mean, in terms of the endpoint titer, antibody binding titer, you can see after each booster up to 3, you can see increase of the binding titer for the monkeys, also pretty good. And when you look at neutralization data, which is against the virus, the autologous virus, that the same sequence as the immunizing antigens. So now you see, you can split into the human study at least. There are some patients, some volunteer, they can elicit pretty good prolonged immune response. Some the immune response, the neutralizing antibodies decline rapidly, and some don't really have very high response. Overall, we see about 75% of the vaccinees can elicit neutralizing antibodies against the virus with the same sequence of the antigens. For monkeys, we immunize 4 of them. We see 3 of them achieve production of neutralizing antibodies. One of them kind of transiently, but never achieved the prolonged neutralizing antibody productions. Then we go look into the antibody response a bit more in detail. First, look into how it binds to different HCV antigens, like different genotypes, with a native, long-reduced, and also a delayed E1, E2. You see overall the human samples that neutralize the virus a pretty good binding cause reactivity against different genotypes. Of course, genotype 1 is better binding. And monkeys, the 3 of them neutralize, it binds pretty well. And because it has higher titer, because we immunize animals with more antigens and also 5 times, I think achieve higher antibody response titer. And if you look at that, it binds to the native E1, E2 a bit better than the reduced E1, E2. So recognize some of this discontinuous epitope. However, when you look at the virus neutralization, it neutralizes autologous virus well. However, but it doesn't cause we neutralize the other genotypes too well. You see some neutralization, but it's nothing impressive. And this is SA13 is a 5A virus, which is known to be very sensitive to neutralization. And the anti-serum can actually neutralize as well as the autologous virus as well. So recently, a number of us grouped together, want to develop better HCV reagents for vaccine studies. This study led by Justin Bailey and Stephen Fang. So we look into a panel of viruses with diverse sequence, and we done a big neutralization study against some of the quite well-known cross-neutralizing antibodies in the field. And we do notice some viruses are more resistant to the neutralization by these antibodies. Some are more sensitive. So kind of roughly divide into 4 tiers, tier 1, 2, 3, 4. And the tier 1 is more sensitive virus to neutralization by this antibody panel. So we took advantage of the human samples, a bit of the leftover. We don't have a lot left. But if you look at that compared to the one I showed you earlier, this human sample, the one we have, it can neutralize the tier 1 virus, which is sensitive virus, pretty well. But again, once you mix with the more resistant virus, you don't see very much neutralization in most of the sample. The same for the monkeys as well. And now we also look into the specificities of the antibodies. Here is a PEP scan assay, look into the antibodies bind to a range, the overlapping peptides across the E1, E2. As expected, you see a lot of reactivity to the XBR1, the hypervariable region 1, and N-terminal of the E1. But overall, they are quite similar between the humans and the monkeys as well. And based on that, we can't really explain the neutralization. These are not neutralizing sera, and this neutralizes the best. Based on PEP scan, you can't tell the difference. But when you look at the more like the discontinuous epitope by competition ELISA, you do see the sample that can block binding of CDH1, which binding to also compete with the AR3 antibodies group. They usually have a better neutralizing activity. And the ones that could not neutralize are the ones that are not making this type of antibodies. And the same for the non-neutralizing sample of the monkey. It doesn't make the CDH1 blocking antibodies. So overall, from the human studies, this analysis, we know the E1, E2 itself are immunogenic, and it can elicit neutralizing antibody response against autologous and also sensitive virus. However, it seems to be not very good against more diverse virus, and it's a bit uncertain whether we can push the antigens itself further to produce cross-neutralizing antibodies. Also, when we analyze the human with the monkey antibody response together, we see they're quite similar to each other in terms of neutralizing activity and what virus they can neutralize, and also the epitope they're binding to. So I want to move on. The next one topic is the challenges to develop vaccine antigens based on the E1, E2. So we did quite a lot of work with these HCV envelope glycoprotein. And here are at least 3 of the main things about when we want to design vaccine antigens based on this E1, E2. And the first thing is the HCV envelope. We know that it's antigenically variable. If you just try to compare with HIV1, at the base amino acid to amino acid level, HCV actually have a higher mutation rate than HIV. Of course, HCV doesn't have as many insertion deletions and drag can compare HIV. However, itself is extremely mutated or variable virus. So this is a problem in vaccine design. And the other part is when one wants to make a HCV antigens for vaccination, itself is actually quite difficult. I just want to show you some cases here. If you try to make the E2 atoll domain by just cloning the sequence and then make a stable cell lines to express this protein. And in our case, we do first purify the supernatant from the stable cell line using antibody affinity column select for folded protein. And after that, you can do a size exclusion and then run the gel here. Here show you 4 different genotypes. And you see they vary quite a lot. Some of those make better monomers, and most of them still have this aggregate of protein, which contain the folded epitopes to be purified alongside with it. And some got pretty poor monomer productions. And these are the covalent aggregates of the protein. And they're always there, and most likely they're in complex with other misfolded protein. So the question really is those contaminants, will that affect in a vaccine when you produce it? On the other hand, when one look into the E2, it's been a long way now. We can do a lot of engineering by truncating some of the regions of the protein and mapping for the binding antibodies. So doing a lot of manipulation in the past, we can delete chunks of the sequence that are flexible. And then we were able to actually make pure enough protein to solve the first structure of the E2 core domains. And here show you. And the problem is when you try to crystallize or solve the structure of the E2 by itself, normally it doesn't work. Because on the protein, it has a lot of regions that are extremely flexible, including the so-called E2 neutralizing phase, which bind the CTH1. If you take the structure, just do an MD molecular dynamic stimulation, you can see this area here is extremely flexible. The gray is the soft structure, and then these regions just keep moving around on the recombinant protein. So when you make the protein for as a vaccine antigen, for immunization, you can expect the host animals or the vaccine is going to see different presentation of the epitopes. And this one we know is for neutralization, but we don't know the other one. So this should be considered in the vaccine design. And indeed, you can even see protein flexibility around the neutralizing phase of E2 by EM. Here just show you the E2 core protein bound by 3 different fat fragments, AR1B, 1A2, and DXC1, which is on one of the bodily neutralizing mechanisms. And you see the angle between this fab, they are quite variable. Some almost like a person jumping, and then the other one look like a person standing with the 2 legs quite close by. When you start to average this protein image, EM images, the 2D class, you start to see one of the legs disappear. It seems the results just suggest that even the neutralizing epitopes on the E2 core is moving around, and on EM you can just see it when you look at different class. So the question is when you have the vaccine antigen with all these epitopes moving around, would they be a good antigens, or you may need something more conformation constraint into the epitopes recognized by the bodily neutralizing antibodies. So for the E2, E12, it has a lot of problems in terms of trying to produce the protein in high yield and good quality. And whether those misfolded proteins come with the purification may cause trouble when you do immunization. And these are the issues we need to deal with in vaccine design. So next one, I want to talk about the neutralizing epitopes on E1, E2. So classically, when I first started, we isolate a panel of human antibodies, and then we map them, and by competition ELISA, during that exercise, we call there are 5 non-overlapping antigenic regions on the E1, E2 complex. And AR3 is the one that overlap with the CD81 binding site, the receptor binding site on the protein. And later on, we kind of start to look into it by overlaying the EM data of the recombinant protein with the structure we have. And then also kind of predict where the E1 is going to be present on the E1, E2. So more recently, Rutger Sanders and Andrew Ward, they actually solved the E1, E2 complex by structure by cloud EM. So their paper is still under review. It's currently available on the bioarchives. This one, I just want to show you the main thing is like E2 and E1 seems to be forming these elongated structures on here. And the AR4 turnout is still around the E1, E2 interface. But the binding of the AR4 actually is only require E2, but you have to fold in a specific conformation for it to bind If you just express the E2 active domain AR4, it will not bind. And on E2, I mentioned, we know there's a large surface area that are accessible from body neutralizing antibodies, and which combine two areas. One is called front layer, the other is called CD81 bind loop, come together to form the discontinuous antigenic surface. And in the middle of it it's relatively hydrophobic for the recognition for neutralizing antibodies. Indeed, many groups, including ourselves, the AR3 series of antibodies, and by Justin and by different groups and Stephen, we isolate many antibodies that are recognizing this E2 neutralizing phase. It is just some of the examples. Some bind to this whole surface area, some bind to more selected regions here, and some bind to this little bit of the so-called AS412, the one moving around on the surface. One of the things that most interesting when you look at these antibodies, many of them are actually belongs to the VH1 using the human heavy chain variable domain VH169. Here just show this group of antibodies. So, and which is pretty interesting. So we start a collaboration with Stephen to look into this VH169 antibodies more carefully by studying the binding and the structures. And when we look at that, with more antibody sequence and structures, one thing we know, as I mentioned, on the E2 neutralizing phase, there's a hydrophobic patch in the middle. And in the VH169 germline genes, the CDR H2, the loop 2, usually have a pretty hydrophobic residues embedded in the germlines as well. That for many of these body neutralized antibodies used to interact with the hydrophobic surface on E2. And usually these antibodies have a longer than average CDR3 loops. And binding most of the time is dominated by heavy chains. Some of the antibodies you only require antibody heavy chain to bind. Light chain is not involved in the binding. And one good thing is these antibodies usually they don't have many somatic hypermutation like the HIV body neutralizing antibodies. So this antibody seems to be achievable when you go vaccinate by usual standard method, and those hypermutation rates should be achievable by vaccination for this VH169 like body neutralizing antibodies for hepatitis C virus. So in the study, we look into here, just show the structure we already saw with the antibody fragment bind to the E1, E2 in this way. And when we solve more structure of Stephen's antibodies, and now we see there's a different way of the antibodies binding to the E2 antigens. And the flipping slightly different from each other. And this is become look like we are looking into two way of bindings of antibodies. However, the way it binds still using quite similar, the heavy light chain, the CDR loops, the way they interact to it, and depending on the CDR, the hydrophobic CDR2, H2 of the VH169 antibodies. However, when we look at the antigen side, the E2 core, we now found that the front layer of the E2 is actually different. One of the group we call the so-called conformation A, we have the front layer recognized in this way, going across the protein and bind to this group of AR3-like antibodies. But when we look at some other antibodies, for example, 212 antibodies, it binds to a conformations a bit different from what we see with the one we saw many times in the past. Now the front layer seems to be somewhere moved like here, away a little bit further in this upway. And in the meantime, because of that, expose some of the residues usually being covered in this conformation in the back layers of the E2. So now it exposed some of the residues. We always thought when you do mapping, when you mutate it, it always lose binding to CD81. Now it's kind of really explain like this surface area here is kind of moving around, may facilitates the binding of the receptor. And also the back layers of the E2s can be involved in antibody neutralization. So I want to go back to the humans and monkey study I mentioned to you earlier. So at that one of the study in here, the importance I think for me is in the vaccine human study, there wasn't any cells left for analyzing the antibodies at the monoclonal cellular level. So when we do the monkey study, which provide opportunity, look at that more carefully. So in this case, we harvest the cells, we isolate the plasma plus and generate a panel of monkey antibodies. So we clone about 100 of those monkey antibodies from the plasma plus. And from here, you can see it have pretty wide coverage of different VH genes being used to make the monoclonal antibodies. But one thing strike out you can see is the one that have cross-neutralizing activity is the VH1.36 class of antibodies. And when you compare the sequence, actually, this is one of the monkey antibody genes closest to the human VH169 germline genes, which was a bit surprising to us. Wow, in monkeys, the monkeys use a human orphomer of the VH169 to make cross-neutralizing antibodies. When you compare these antibodies, the VH genes together, you can see the human antibodies from here, they all form the bases, the branches. They are almost equal distance to these monkey antibodies when you do the phylogenetic tree. So they're very closely related. However, when you study how these antibodies recognize E2 for neutralization and solving the structure, now we see something interesting. So the one I mentioned to you, like the classical AR3 binding, you have the E2, you have the heavy and light chain of the fat fragment come in this angle, and this mode of binding. And when you look into the how the monkey antibody bind, now you see the antibodies kind of switch with their light chain swap onto the other side to interact with the core domain. Okay. And also one thing interesting to us is, if you look at how all these human antibodies bind, the hydrophobic CDR2 usually come closer, sit more in the middle, interact with the hydrophobic patch of the E2 neutralizing phase in the middle of it. For the monkey antibodies, actually kind of interesting. Come on the side, now the CDR3 actually more occupies in the hydrophobic area for the monkey antibodies. So we, in this study, we found like, okay, monkey use overall antibody genes to produce antibodies bind to the same neutralizing surface, which is interesting. And they also react or cause neutral HCV. However, the way it recognizes antigen surface is very different. So at this point, really can't be certain, is it real? Is it a coincidence? Or actually the functional and the genetics conserve in this case? And this one actually kind of gives some interesting insight. And last month, the group, the Florian Klein group in Germany, they published these studies, which is extremely interesting to us because it can't really come back to the whole thing. It's what we saw in monkeys also happening in humans. So they go to study, they study a number of HCV elite neutralizers and analyze the neutralizing antibodies at the monoclonal level. In the same way, they found a lot of the neutralizing antibodies belong to VH169. And when they look into more carefully of those antibodies, here just show some of the examples, like the one they isolate from patients compared to those also isolated from patients by the single B cell method or by recombinant methods. And they all have this cross-neutralizing activity to it. The most striking part is now the antibodies, the way it bind to it, actually more like the monkey's antibodies than what we classically see in humans. And you can see in here, the way it bind, it recognizes in this angle, and then the footprint of how the antibodies bind is almost like the monkeys. And of course, the most strikingly, you see, I explained to you how CDR2 bind with these classical antibodies. It come in around this, try to interact with the center of the E2 neutralizing phase. And now you look at the new antibodies isolated by the Klein group, you see the way of the approach of the antibodies, almost like the monkeys. Like the CDR2 is on the top instead of on the bottom. And you can even kind of tell where's the light chain here. In this case, the light chain is on this side, and you can see it classically, because of the switch of the antibodies binding mode, it comes from the other angle. So it's really kind of interesting. We found from monkey first a new way of VH169 antibodies neutralize HCV, and then later on we found in humans as well. So because of all these information all come together, there's really a so-called multi-donor class of body neutralizing antibody response when people see the HCV E2 antigens, and some people call it public neutralizing antibodies response. and even more interesting seems to be this class of antibodies that conserved in humans, also in non-human primates. So to summarize a bit about the cross-neutralizing epitope of CV for vaccine design, we know the E2 neutralizing phase is quite immunogenic. Many antibodies been isolated to it. I mentioned the AL4. Now the structure is out. We can think a bit more how to design vaccine based on that. And there's a multi-donor class as a neutralizing antibody response. And seems to be the non-human primate, the macaque model will be very useful to study this type of neutralizing antibody response. Okay, to finish with my talk, I just want to quickly talk about like some of the strategy with the structural information, with this binding mode, with this multi-donor class of antibodies. One of the key issues, really, we need to think about how to stabilize and better present the neutralizing epitopes. And one thing we didn't talk a lot about, but it's quite essential in terms of vaccine design, you really need to make antigens have a very, very, very favorable properties, good antigenicity, good yield, purity, and stable for vaccination, and easy to produce. And some of the approved human adjuvants, we already tested CPG. It also worked with E2. We don't know about the LNP that combined with the mRNA vaccine, which now known is also very immunogenic. So the way to improve the immunogenicity, one of the approach we take is try to first optimize our antigens and put onto these self-assembling protein nanoparticles, championed by my colleagues Jiangsu and Scripps. And the idea pretty much, this is a one single protein. It will express in high yield and can be produced at the GMP level. And will cell assembly relatively easy to produce and form nice nanoparticles. And he has some data to show this nanoparticle are more stable and retain into the lymph node for a while, and then much longer than just a single protein. And it can elicit germinal center B cell and TFH cells much faster than these monomeric protein in comparison for the protein displaced on the nanoparticles. So we know that we can displace E2 on some of this nanoparticle. And now we kind of see continuous optimizing the protein, also see whether we put E1, E2 on top of this nanoparticle and to produce it. So I want to conclude my talk, and we have teams at Scripps Research also with Chris Chen in the Southwest National Primary Research Center to push this HCV vaccine effort using the approach quite different from the others existing one. And I also want to thank for many people in the field and help our efforts and being an extremely good collaborator. Also want to acknowledge the funding from NIAID and NIH to support our work. Yeah, thank you.
Okay, great. Thank you, Manson. That was a terrific talk. I think in the interest of time, we will move to the next talk, but there's some terrific questions in the Q&A. And Manson, I'm not sure if you could get a chance to answer those in the Q&A and chat function. And then we'll also have an opportunity during the panel discussion to address, I think, some of the issues that have been raised. So Manson, thank you again.
Yeah, sure. Yeah, I'll answer those questions on NIAID.
Okay, great. Thank you. Our next speaker is Justin Bailey from Johns Hopkins University. The title of his talk is HCV Vaccines and Neutralizing Antibodies, Lessons from Human Infection. Welcome, Justin.
Thanks, Chris. And thanks for the invitation to talk today. Thanks also to Rajan and the other organizers of the conference. So it's my pleasure to follow Manson. Always a hard act to follow, but I'm going to talk a little bit on some of the same topics, but from quite a different, I think, perspective. So the first question that I think there's a consensus now on this question, but should a vaccine induce neutralizing antibodies to protect against hepatitis C? I think that most of us think that the answer is yes at this point. That wasn't always the case. But a lot of that consensus, I think, came from data acquired from natural HCV infection. And so I'm going to talk first today about polyclonal neutralizing antibodies and their role in spontaneous clearance of hep C. talk a little bit about human monoclonal antibodies, and I think most importantly, talk about the fitness cost to the virus of resistance to these antibodies. And then I'm going to come back at the end a little bit to talk more about that pseudoparticle panel that Manson introduced in his talk, that we developed collaboratively across multiple research labs, that I think is going to be a really important tool for the field in general. So all of the work that I'm talking about today is made possible by the BBASH cohort, which is a cohort that was established at Hopkins about 20 years ago now by Dave Thomas, and then maintained and enhanced by Andrea Cox since then. This is a group of people who inject drugs who are followed prospectively from before the time they're Hep C infected. And then despite counseling and other support, some of them unfortunately do acquire hepatitis C infection. And they're followed monthly over the course of that acute infection until the time that about 25% of people spontaneously clear hepatitis C without treatment, and 75% go on to develop persistent or chronic infection. And so, of course, this gives us an amazing opportunity to study the immune responses that are present at this critical junction during acute infection, when some people are clearing infection and others are developing a persistent chronic infection. The other thing I'm going to refer to a lot today, and others have talked about this as well, but what is an HCV pseudoparticle? So just for those who don't know, you actually can co-transfect an HCV envelope protein expression plasmid with a defective HIV or MLV backbone with a reporter gene. And what you get is a particle with an HIV core with the reporter gene, but with hepatitis C E1, E2 proteins on the surface. And these particles do enter hepatoma cells in a receptor-mediated way, and you can measure quantitatively neutralization, for example, by antibodies that block that entry. So using this pseudoparticle technology, clear back in 2014, Bill Osborne and Stuart Ray in my group developed one of the first diverse genotype 1 pseudoparticle panels, which is depicted here on the left. And they used that panel to measure neutralizing breadth of plasma in people in the BBASH cohort who were either about to go on to persistent infection or those who are right on the verge of clearing their hepatitis C infection. And these two groups were time matched for duration of infection. I think that's a very important thing to know. And what they saw was that in these time matched samples, the people with clearance of infection did have greater plasma antibody neutralizing breadth on average than the people who went on to persistent infection. Now, there's some interesting things to notice in this graph. First of all, there's a group down here that did not have detectable heterologous neutralizing antibodies at all, but nevertheless spontaneously cleared their Hep C infection. And I think, given other data on the importance of T cells in clearance, these may well be individuals who cleared primarily through T cell immunity. There's this group up here, which are people who did develop broadly neutralizing antibodies very early in infection and then went on to clear infection. And we pretty commonly now call these elite neutralizers. There's also this interesting group, this troublesome group of people who also early in infection developed broadly neutralizing plasma antibodies and nevertheless did not spontaneously clear their HCV infection. So one of the things that we're really interested in understanding now is what's the difference between these people with high NEUT who clear infection and these people with high NEUT who did not clear infection. This particular issue is even more, I guess, confusing, you could say, because broadly neutralizing monoclonal antibodies have been isolated both from people who cleared HCV infection and also from people with chronic HCV infection. And so what I've shown here is a crystal structure of the E2 ectodomain. and marked on it some of the most commonly targeted antigenic sites on E2 by various different broadly neutralizing monoclonal antibodies. A lot of these were isolated in Manson Law's lab, in Stephen Funk's lab, some by me and Andrew Fliak in collaboration with James Crow at Vanderbilt. The monoclonals marked with asterisks were isolated from people who cleared infection, and the other monoclonals that are shown were all isolated from people with chronic HCV infection. And so one thing I'll point out is we did recently isolate this monoclonal that we called Hep C108 that does appear to target a distinct or unique antigenic site relative to the other BNAbs that had been described. And it was isolated from someone who spontaneously cleared HCV. And I'll just use that antibody to make the point that I think that we've still really only scraped the surface of understanding the different neutralizing epitopes that are targeted by these different antibodies. You know, we tend to lump them into antigenic sites for convenience, but it's important to notice or to note that each of these monoclonal antibodies targets a distinct epitope. So it may be that targeting one particular residue within that antigenic site may potentially make the difference between clearance and persistence. And that's something that we're continuing to study, and I think it is going to be important to understand. The reason I'm optimistic that we're going to be able to make a lot of inroads on that question is this new approach that we have now to isolate E2 specific B cells. Most of the mAbs that were isolated in the past used hybridoma or phage display technology. And Arash Brakui, who spoke yesterday, really showed the first proof that we could use this E2 staining strategy to isolate HCV E2 specific B cells. And we've since developed a similar assay in my lab. And in our particular version of this assay, we actually use a cocktail of antigenically distinct E2 proteins in order to increase the sensitivity of this pull down. And so this has really massively improved the efficiency of both studying E2 specific B cells from infected people and also isolating monoclonal antibodies. Nicole Skinner, when she was a postdoc in my lab, took advantage of this approach to actually study BCR sequences, to do BCR-seq from some of these BBASH subjects. And she actually selected subjects for this study based upon that Osborne study that I was showing you earlier. In that, she took into account both the neutralizing activity of the plasma of the individual and also the outcome of infection of this individual. So rather than having two groups of clearance or persistence, she actually had four groups that she called high neut clearance, high neut persistence, low neut clearance, and low neut persistence. And then she compared the BCR repertoires of these different groups. And the really fascinating thing she saw was that there are public B cell clonotypes within these groups. And her definition of a public clonotype were BCRs that shared both V and J usage and had identical CDR3 sequences. And the most sharing, or the largest number of public clonotypes that she picked up were between two individuals from the high neut clearance group. And these were public clonotypes that were not found at all in the high neut persistence group, for example. So when you look at this, high throughput and really high resolution, you start to see that there really may be differences in the monoclonal antibodies that are generated in people who spontaneously clear Hep C infection compared to those who don't. Even if looking at the plasma, it looks like both plasma samples are relatively broadly neutralizing. As a control, of course, she looked at the E2 non-specific B cells. And interestingly, the clustering here, or the sharing of public clonotypes, looked to be largely driven by the genotype of virus that the person was infected with. And so, we'd speculate that these may be antibodies that target non-structural proteins rather than envelope proteins. So the next question, Arash Barkui brought this up yesterday when he spoke. What we're really looking for, I think, is direct evidence that broadly neutralizing antibodies play a role in clearance of infection rather than just correlations like I showed you with the Osborne study. And so what we've done is follow both the viral evolution and the neutralizing response in people who are either clearing HCV infection or developing persistent infection. And we've done this now in quite a few people. I'm just going to show you one person as an example for simplicity. So this is a subject that we call C117 who spontaneously cleared HCV without treatment. Actually was infected for about a year prior to clearance. And what we did was we measured the plasma neutralizing activity over that course of infection. And we also, with the help of George Shaw at UPenn, did single genome amplification of the virus that was present throughout the course of infection in this person. And with the help of Andrew Fliak and James Crow, we isolated also broadly neutralizing monoclonal antibodies from the B cells of this person right after they had cleared infection. So this is just to show briefly that this person did develop broadly neutralizing plasma over the course of their infection. And by day 388, immediately prior to clearance, they did neutralize the majority of HCV-PP variants in this panel of 19 that we were using at the time. This phylogenetic tree shows the evolution of the E1, E2 genes over time in this individual. And these are color-coded by the time of sampling. So you can see that there was sequential evolution of the virus over time in E1, E2. And they're really fast, and you see these tight bottlenecks as well over time, implying that there was strong selection. And the really cool thing is by day 388, immediately prior to infection, there was only one remaining clade of virus, all these viruses that were very closely related to each other, differing only by a few nucleotides. All of these other variants that had preceded were already cleared, and the virus was just hanging on with this one last clade of virus. Then we looked at where the substitutions were showing up. This is the 5' hemigenome of those viruses. Where were the substitutions appearing? And it turned out almost all of the substitutions were showing up and becoming fixed in the E2 protein. Some were in the hypervariable region, as expected, but a lot of other substitutions were showing up in the more conserved regions of E2, including, this is actually the front layer of E2, which is the CD81 binding site that Manson was describing in his talk. So I mentioned that we had isolated a BNAb, a monoclonal from this person that we called HepC3, and Andrew Fliak in 2018 in Pamela Bjorkman's lab was able to crystallize the FAB of this antibody in complex with an unmutated E2 ectodomain to show us exactly where this monoclonal antibody was binding. And overall, the binding was quite similar to what Manson had seen with his AR3 antibodies, and it was to the front layer of the E2 protein. So what we're showing here are just the critical binding residues of this HepC3 antibody on the E2 protein. And we're comparing it to the critical binding residues for the CD81 receptor on the same E2 protein. So you can see that there's an amazing amount of overlap in the critical binding residues of the mAb and this CD81 receptor. So this led us to hypothesize that escape from this antibody may have carried fitness cost for the virus. So the first thing that we did was we looked at whether the changes that appeared in the envelope protein over time did confer resistance to autologous antibodies. So here on the top graph, we're showing binding of plasma antibodies to these sequential E1, E2 proteins. And you can see that the later E1, E2 variants are more resistant to the autologous plasma antibodies than the earlier E1, E2 protein. We just introduced, or then we tested the same thing with this Hep C3 monoclonal antibody, and you see the exact same pattern, suggesting that Hep C3-like antibodies were responsible for a lot of the neutralizing activity or binding activity we were seeing in the plasma of this person. So then we looked at fitness of these longitudinal viruses. Just using pseudoparticles, we could see a significant drop in the ability of these E1E2 proteins to mediate entry over time, about 100-fold drop in pseudoparticle entry. We also cloned these E1E2 genes into replication-competent virus to make chimeric HCV-CC and measured the replication capacity of these viruses. And again, you can see that the early virus is quite fit and able to replicate in vitro. But by the time you get to that last virus that was present immediately prior to clearance, the virus is completely replication incompetent. We wanted to figure out why that happened. And so we measured the capacity of these longitudinal E2 proteins to bind to either CD81 or SRB1 that was expressed on the surface of CHO cells. And so when we looked at CD81 binding, we see a pattern that's almost identical to what we saw for viral fitness, which is the early E2 bound quite well to CD81, but the later E2 prior to clearance bound very poorly to CD81. And notably, that was not the case with SRB1 binding. And then when we just introduced substitutions in the more conserved regions of E2, ignoring the substitutions that appeared in HVR1, we also saw the same pattern. We saw this loss of CD81 binding as we introduced those substitutions that happened naturally. And again, saw no effect on SRB1 binding. So again, that was just one person. That paper, we described the same thing in two individuals. We've subsequently done this work with a lot more cleared infections. And a lot of these infections shown on this graph were actually people who repeatedly cleared multiple infections. And interestingly, we saw exactly the same result, which is the infections that were cleared, when we compared the early envelope protein to the late envelope protein, we saw this drop in CD81 binding. But when we did the same thing with infections that were not being cleared, compared early to late E2, we saw no decline in binding to CD81. So to put this together, I think this is strong direct evidence that neutralizing antibodies are playing a role in natural clearance of HCV infection. And it's not because they're amazingly potent, perfectly broadly neutralizing antibodies. It's just that they're potent enough and broadly neutralizing enough and target conserved epitopes to the point that escape from these antibodies confers a pretty significant fitness cost to the virus. So, this is an illustration of, driving bison off a cliff to kill them rather than shooting each one individually. And I think that that's an analogy of what's happening with BNAbs in HCV infection. So with the time I have available left, I'm going to talk a little bit about this standardized pseudoparticle panel that, again, Manson introduced. I think that this is really, really important for the field for us to start to use a standardized panel so that we can compare neutralization results between different labs and different vaccines. So to develop this panel, the first thing we did was we looked at all worldwide HCV E1E2 sequences, and we picked out E1E2 clones that we had available and mapped them onto this tree, and selected the E1E2 clones that we thought were most representative of the worldwide sequences. And this turned out to be 75 different genotype 1 to 6 E1E2 clones. When we made pseudoparticles, 65 of these clones produced functional pseudoparticles that we could use for neutralization assays. This is the second time you've seen this graph, but we saw when we tested neutralization of a set of 6 different monoclonal antibodies that there was a wide range of neutralization sensitivity of these different pseudoparticles. And one thing I'll point out specifically is that there was no relationship between the genotype of the pseudoparticle and whether it was neutralization sensitive or neutralization resistant. So the non-genotype 1 PPs were scattered across these neutralization sensitivity tiers. Rather than just looking at the mean or the median neutralization sensitivity of these pseudoparticles, we also looked at the pattern of neutralization of each pseudoparticle by all 7 monoclonal antibodies. And we clustered the different E1, E2s based upon their pattern of neutralization by these 7 different monoclonal antibodies. And the really cool thing is that all of these 65 E1, E2s clustered into 15 different groups of very antigenically similar E1, E2 proteins. And some of these groups included E1, E2 proteins from multiple different subtypes. So the clustering, the antigenicity of these E1, E2 proteins was not dictated by their genotype. Using all of this data, we were able to narrow this panel of 65 pseudoparticles down to 15 representative pseudoparticles. And when we look at IC50s or neutralization breadth measured with either the panel of 65 pseudoparticles or the smaller panel of 15, we get almost exactly the same result. So we think it's not necessary to test every plasma sample or every antibody against 65 pseudoparticles. Instead, I think that we can test against these 15 representative isolates. And importantly, when we tested neutralization by immune sera, these 15 different pseudoparticles, we see the expected ranking of neutralization resistance that we saw with the monoclonal antibodies that we used to select the isolates. And that's with sera from patients infected with genotypes 1 through 6 HCV. You still see this same ranking of neutralization sensitivity. So to summarize, I think bNAbs play an important role in clearance of HCV infection. Clearance appears to be associated with antibodies that can drive resistant substitutions that reduce E2 binding to CD81. E1, E2 antigenicity is not dictated by genotype. I think that's a really important point, both for neutralization testing, but also for selection of vaccine antigens. And last but not least, this pseudoparticle panel I've described is freely available. You can get it from AdGene. You can ask me directly, and I'm happy to share it. So I want to thank my team who's done most of this work. I want to specifically point out 2 people. Nicole Skinner did some of the work I showed. She recently left my lab, unfortunately, and has started her own lab in Chris Walker's group. So a rising star in the field. And Andrew Fleak, that you've all heard of before. Believe it or not, he has not been a PI all these years. He's been a postdoc in Pamela Bjorkman's lab, but he's now starting his own lab in the next few months at Cornell. So keep an eye out for both of those folks. And last but not least, thanks to NIAID for funding all of this work through the U19 and various R grants. Thank you.
Great. Thanks for that talk, Justin. That was fantastic. So we have time for 1 or 2 questions. We have one in the chat from Dr. Raju. Is there any source or database to get known bNAbs of HCV is the question.
That's a great question. Not that I am aware of. We've written a few review articles. Stephen Fung has written a few review articles. Manson's written a great review article that talks about some of these bNAbs. So I'd point to those, but I don't think that there is a database.
Okay. Justin, I'll ask a question. It appears that there are a number of people that just don't make a good neutralizing antibody response. Some ultimately clear, some don't. But is there something different about those viruses or other aspects of other components of the immune response, the helper response? I don't know if you've sort of been able to integrate that type of analysis, but why they don't respond.
Yeah, no, that's a great question. So we've looked a lot at this, and I think there are two things that we've seen. First is that the phenotypes of the B cells are different in people who develop good antibody responses and people who don't. Now, why that's the case, we don't know yet. But we just published a paper recently in PLOS Pathogens where we showed that expression of FcRl5 and PD1 on the surface of B cells was associated with a poor E2 specific antibody response. And then the other thing I'll mention is that we've been doing work lately in this reinfection cohort to actually characterize the envelope proteins in people who developed broadly neutralizing antibodies and people who did not. And there do seem to be antigenic differences in the proteins that these people are exposed to that associate with whether they develop BNAbs or not. And that's a paper that's in review right now.
Okay. Thanks, Justin. Seeing no other questions in the Q&A,
Chris, could I ask one question?
Yes, Jens, please go ahead.
Guys, just that, Justin, great talk. If you think that the role of broadly neutralizing antibodies in clearing HCV is to drive the virus over the cliff, how can we reflect that in our in vitro neutralizations assays?
I would say we just have to make our best guess. And so, what we've done with this new pseudoparticle panel is we've measured neutralizing activity of plasma samples of people who cleared and people who did not. We'll publish those data. And I think that can be a reference that immune sera could be compared to see how closely they simulate what we see in people who are clearing. If there were an animal model where we could look for development of escape mutations, then that would be another possibility to actually sequence the virus over time and look for development of these substitutions in the CD81 binding site of E2.
Okay, I think we have time for one more question from John Law, the University of Alberta. Do you know the quality of the antibody response in people who do not clear the virus?
Yeah, so it's widely variable. There are some people that have what look like really great broadly neutralizing plasma antibody responses who do not clear. We think that in those folks, one, that they may not be targeting exactly the same epitopes as people who clear, and two, that they may be infected with a different virus that's more able to escape and compensate for escape mutations than virus of people who do clear infection. And so those are things that we're studying now, but we don't know the answer yet.
Great. Thanks, Justin. With that, then I'll turn the session over to my co-moderator, Georg Lauer, to introduce the next talk.
Thank you, Chris. Our next speaker is Bali Pulendran from Stanford University. Dr. Pulendran has really pioneered the use of systems biology approaches, to get a holistic understanding of how innate immunity unfolds after vaccination and then how it shapes afterwards adaptive immune response to the vaccine. And he's gonna help us understand today how we could use this for hepatitis C virus vaccination by his latest work on the Pfizer-BioNTech coronavirus vaccine. Thank you for joining us, Bali.
Great. Thank you very much, Georg. Can you see my slides?
Yes, we can.
Perfect. Okay. So I'm really delighted to be here at this meeting on HCV. As many of you know, we don't work on HCV, although in recent months, we are delighted to be involved in a very exciting U19 NIH-funded program headed up by Stephen Fowle and working together with Stephen and Chris and Mike and Richard. So we're learning. We're very much learners. But we're looking at this from the perspective of really of understanding immune responses using the systems biological approach that Georg mentioned. And so what I'd like to tell you about is what we are learning from using the systems biological approach about immune responses to COVID vaccines and how we think some of those lessons might be instructive in the case of HCV. All right, so let me spend a few minutes telling you about some published data from this paper that was published a few months ago. And this was really a study that we undertook right at the beginning of last year when the Pfizer-BioNTech vaccine was being rolled out. Of course, this was a very exciting new platform, the first of its kind. And we and many others were keen to understand immune responses to this vaccine in humans. So together with my colleagues at Stanford, we performed a clinical study involving 56 healthy adults who received this Pfizer-BioNTech vaccine, 2 shots, as you know, day 0 and day 21. And our goal was to perform an extensive analysis of the innate and the adaptive response to vaccination by analyzing blood samples obtained at these various time points. And we used a variety of different techniques shown here, some of which I'm going to show you data from. Well, what about the neutralizing antibody response? Here we're looking at live virus neutralizing antibody response. As we now know, these mRNA vaccine platforms, both the Pfizer and the Moderna platforms, are really quite efficient at inducing robust neutralizing antibody titers. As you can see here, it really does take 2 immunizations to get a high enough titer. 1 immunization doesn't quite do it. And in our study, we found that both males and females developed similar magnitudes of neutralizing antibody titers. Interestingly, this Pfizer-BioNTech vaccine also seems to induce a substantial antigen-specific T-cell response. What we're looking at here is ex vivo stimulation of PBMCs isolated from these vaccinated subjects, stimulated ex vivo with overlapping peptides from the spike protein, and then doing an intracellular cytokine assay to analyze the cytokines produced by these T cells, including interferon gamma, TNF, and IL-2. And on the left, you see the CD4 T cell response, and on the right, CD8 T cell response. Now, these frequencies may not look all that high, but they are higher than what other vaccines have been shown to induce. I'm thinking here about inactivated vaccines. Remember, this is an inactivated vaccine, not a live viral vector such as yellow fever, which really induces a very high magnitude of CD8 T cells. Nevertheless, you can see at day 28, which is 7 days after the second immunization, a pretty respectable magnitude of antigen-specific CD8 T cells in the blood. Now, we also looked at the innate response, and one way in which we looked at the innate response is to analyze the change in gene expression profiles induced by vaccination in the blood, in total peripheral blood mononuclear cells. And if you focus on the left panels, we're looking on the y-axis at DEGs. DEGs are differentially expressed genes in response to vaccination. So you can see on the left panel, the left panel is what happens after the primary vaccination. And on the middle panel, you see what happens after the booster vaccination. And the main point I want to make here is that booster vaccination appears to induce an enhanced transcriptional response relative to the corresponding time points after the primary vaccination. Now, if you look on the right heat map at what exactly is this transcriptional response, you can see that we've done some analysis of the transcriptional modules, and you can see that these are modules that are characteristic of the antiviral innate response. You can see, for example, the antiviral interferon signature, type 1 interferon response, viral sensing in IRF2, and so on and so forth. So the main point here is that this vaccine is inducing a robust innate response. Interestingly, though, it seems to be a heightened innate response after the secondary immunization, much like a heightened T cell response or a heightened B cell response. Well, to dive into this in a little bit more detail, we did some single cell transcriptional profiling using this technique that we call CytSeq. And here's a UMAP showing an example of the different cell type clusters that you can define using the site-seq approach. And you can indeed define many different subsets of lymphocytes and myeloid cells and DCs and so on. I'd like to make one point on this site-seq analysis, and that point is shown here on this slide. What you see in these boxes are the UMAPs that I had, the UMAP that I had shown in the previous slide. Now you just see these clusters in gray color, but I'd like to draw your attention to these this cluster of red cells here. You see there are very few red cells at day 0. Upon vaccination, you see an increase in the frequency of red cells at day 1, and this is apparent at day 2, disappears by day 7. At day 21, the day of the secondary vaccination, there aren't any, but look what happens immediately after the secondary vaccination. There's a dramatic increase in the frequency of this cluster. Now you might say, what is this cluster? And this cluster is something that, well, first, before I answer this question, if you put some numbers on this, on the frequency of this cluster of cells, it's in fact a myeloid population of cells, a mixture of monocytes and myeloid dendritic cells. And we're looking at the frequency of these cells as a percentage of all lineage negative HLA-DR positive cells. And you can see after vaccination, there's hardly any, less than 0.1% or so. That inset, you can see it's far less than 0.1%. However, after the secondary vaccination, the numbers rise to levels that approach 1%. So in other words, a 100-fold increase in the frequency of this population of myeloid cells. Now, this is very much reminiscent of the kind of increases one sees in an adaptive immune response with antigen-specific T cells and antigen-specific B cells. Now, what exactly is this cluster and what genes does it express? And this cluster is what we term cluster 8 or C8 on the left panel. As I said, it consists of activated myeloid cells, including activated monocytes and activated myeloid dendritic cells. It expresses IRF8, STAT1, STAT2, IRF1, STAT3. Many of these transcription factors you recognize are involved in orchestrating the antiviral response. However, it expresses much lower levels of FOS, Jun B, ATF3, Jun D. These are the so-called AP1 transcription factors. Why this was so interesting to us was that in a completely independent study with a very different vaccine, in this case, the avian influenza subunit vaccine, H5N1 subunit vaccine, that's administered with this adjuvant known as ASO3 that's developed by GlaxoSmithKline. It's a squalene-based adjuvant. We had done this last year. We published this sometime in the middle of 2021. We saw a virtually identical population of cells to this cluster 8 that we were seeing with this Pfizer-BioNTech vaccine. And this population of cells expressed very much the same transcriptional profile. The kinetics of emergence of this was very similar to what we had seen with the Pfizer-BioNTech vaccine. And more importantly, we had also done single-cell epigenomic profiling. We saw that long after vaccination, several weeks and sometimes months after vaccination, there was chromatin remodeling in this population of cells, such that the transcription, the loci, the chromatin loci targeted by these transcription factors, such as IRF8, IRF9, IRF2, IRF7, and so on, were wide open, even months after vaccination in this population of cells. So this was very exciting, and it hinted at the possibility that perhaps this sort of innate recall response that we had observed might be a more broad phenomenon seen with not just this RNA vaccine, but with other vaccines. Now, curiously, getting back to the Pfizer mRNA vaccine results, what we saw was that when we measured cytokines in the serum using Luminex, what we saw was this phenomenon. So you see on the left panel, we're looking at interferon gamma, and this is after the primary vaccination. You see a little bit of an induction there. After the secondary vaccination, there seems to be a substantially higher magnitude of interferon gamma. Why this was interesting was on the right panel, if we correlate the frequency of this cluster 8 population of myeloid cells that I mentioned to you versus on the x-axis, the concentration of interferon gamma in the plasma, you see a really remarkable correlation. this raised the prospect that, well, it must be this early burst of interferon gamma in response to the secondary vaccination, so soon after secondary vaccination, one day, that must be impinging on these myeloid cells and programming them to acquire this transcriptional profile that we had observed. To test this, now this gets into the realm of mechanistic science. And of course, the human model is wonderful to do descriptive science, but it's a little limited in terms of perturbing the system. So now we transition into the mouse model to dive into this mechanism a little bit more. We were able to get approval from the FDA and from the Stanford School of Medicine to be able to use droplets of the Pfizer-BioNTech vaccine that would normally have been discarded after a day of vaccination to now do experiments in mice. And I'd like to show you 2 slides, and this was published just a few days ago. Much as we had seen in humans, in mice, upon vaccination, we see an initial burst of interferon gamma in the serum at day 1. And then after secondary vaccination, you see a substantially increased concentration of serum interferon gamma. And much as we had seen in humans, in mice, if you look in the draining lymph nodes at monocytes, you see that there's an enhanced expression of these genes that we had identified as being expressed in cluster 8. And some of these are IRF7, STAT2, STAT1, IRF8, and so on and so forth. And as we had seen in humans, some of these AP1 transcription factors, such as CFOS and FOSB, were diminished in the level of expression at 1 day after the secondary vaccination. So at this point, we thought, aha, the mouse model recapitulates salient features of the innate response we have seen in humans, and therefore we might do some mechanistic studies in mice. The first question to ask was, well, this early burst of interferon gamma at day 22, where does it come from? It seems so early. In order to get antigen-specific T cells, it takes a good 7 days or maybe 2 weeks. How can it be that there's this initial burst? And so the obvious candidates were, well, it must be NK cells or innate lymphoid cells, perhaps, which is what we did in panel C by looking at intracellular cytokine staining in the draining lymph nodes. And you can see NK cells certainly produce a high quantity of interferon gamma, but so do a population of T cells, including CD8 T cells and CD4 T cells. And you can see the numbers on the right there. Now, these are not tetramer positive T cells. They seem, as far as we can tell, they do not seem to be antigen specific. This begs the obvious question of what are they? What is a population of T cell that results in an initial burst of interferon gamma so soon after vaccination that then impinges on the subsequent innate response? We don't have the answer to this, but we're investigating whether this might represent a special population of T cells in the lymph nodes that possesses some innate-like function. And then the final experiment we did was to ask, is this interferon gamma induction early on important in the orchestration of the myeloid cell response? So to do this, what we did was to prime with the BioNTech vaccine and then neutralize the activity of interferon gamma by injecting an antibody against the interferon gamma receptor prior to the secondary immunization. And then we analyzed the ensuing innate response shortly after the secondary immunization. And what I can tell you is that there's a substantial abrogation of the activation of innate population shown here as measured by CD86. There's a substantial impairment in the expression of these interferon stimulated genes, the same ones that were present in cluster 8, if you block the activity of interferon gamma relative to if you use an isotype control. Conversely, there's enhanced expression of these AP1 transcription factors, suggesting that this early burst of interferon gamma is indeed playing a pivotal role in orchestrating this heightened, enhanced innate response. And the final piece we asked was, how is this Pfizer-BioNTech vaccine sensed by the innate immune system? It's an RNA molecule. It's inducing tremendous T cell responses and antibody responses. What are the innate sensing pathways? And in the interest of time, I'm not going to show you the data, I refer you to this paper, which was published just a few days ago, but I can tell you, so we looked at mice that are deficient in each of these different toll-like receptors, and the adaptive immune response and the innate response seems to be completely independent of these TLRs. We looked at mice that are deficient in these inflammasome molecules, ASC and NALP3, and the adaptive responses are completely independent of that. They are independent of STING and C-gas. They are independent of the various cell death pathways, including necroptosis, for which we use the RIP3 kinase knockout mice, and pyroptosis, for which we use the Gasdermin knockout mice. However, they seem to be independent. They seem to be dependent on MDA5, which is a cytosolic sensor of double-stranded RNA structures. And you can see the results here. Immunization of MDA5 knockout mice results in a substantial impairment in the tetramer-positive CD8 T cells in the lung and in the spleen. They also result in a substantial impairment in the interferon gamma producing CD8 T cells. However, the antibody response induced by this is hardly affected, even though their statistical significance is really not much of a difference at all. So the mystery of how this vaccine is inducing antibody response remains unsolved, and we're working on this. And how exactly is MDA5 involved? Well, it seems to be through the early induction of interferon alpha. So you can see within 6 hours after vaccination, there's this tremendous induction of interferon alpha in wild-type mice in black, 0 in the MDA5 knockout mice, and consistent with this, there's much less activation of dendritic cells, and consistent with this, in the interferon alpha receptor knockout mice, you find hardly any CD8 T cells. So we believe the vaccine, or some component of it, presumably some double-stranded RNA structures that develop spontaneously, perhaps within the cell, is activating this MDA5 pathway through the interferon alpha induction. So this summarizes what I've basically told you. I'm not going to spend time on this. I just want to conclude by asking, what, if any, lessons can we learn from what the analysis of the COVID vaccines are telling us? So I think the first lesson is to say the mRNA vaccines provide an exciting new opportunity to rapidly test novel HCV, it could be HIV as well, vaccine concepts and candidates. And that's very obvious to all of us. The second issue is that as great as the mRNA vaccines are, they're not perfect, because there seems to be a problem with waning durability of antibody responses. So that's a major challenge. So new approaches are needed. So let me illustrate this. Here is another analysis that we did when we had enough time points to now assess, to calculate, working with modelers to work out the durability or the decay rates of the antibody response to the mRNA vaccine. This is the Pfizer-BioNTech vaccine. You see, whether you look at binding immunoglobulin titers or neutralizing titers, the decay rate is about 56 days. That's not very long if you put this in context, because live viral vaccines induce antibody responses that have a decay rate of 500 years or some ridiculous number, virtually incalculable. It's been calculated by Mark Slifka in a beautiful New England Journal of Medicine paper. So why is it that the Pfizer-BioNTech vaccine seems to fall short in that regard, or even the Moderna vaccine? Well, perhaps a clue might come from the analysis of the innate responses. I mentioned to you that this vaccine doesn't activate TLRs, doesn't activate the inflammasome pathway. It does seem to be activating the MDA5 pathway, but that's only in the case of CD8 T cells. In contrast, if you look at a live viral vaccine, such as the famous yellow fever vaccine, which has been administered to 600 million people worldwide, that induces lifelong protection, we had shown previously that this vaccine was in fact activating TLR2, 3, 7, 9, RIG-I, and MDA5, and in knockout mice in which you could abrogate these TLRs expression, the antibody response and the persistence of the antibody response was completely abrogated. So perhaps that's a powerful lesson. Maybe you need to push the right innate buttons to get durability, which begs the question of, well, what about adjuvants that push the right innate buttons, such as TLR7 ligands or ASO3. So it does appear that certain of these adjuvants, some of which we've published on, do seem to induce quite durable responses. And I just want to show you 2 slides from that. One of which is shown here in the context of the study that we did in non-human primates with the recombinant vaccine against SARS-CoV-2 designed by Neil King. We tested a bunch of different adjuvants. I'd like to highlight one example. This is that ASO3 adjuvant that I mentioned previously in the context of H5N1. We're looking at neutralizing antibody titers in monkeys, day 0 primary vaccination, day 21 secondary vaccination, really quite high titers, 5,000 or so. And you see the decline is not all that bad over a 6-month period, about a 7-fold decline. And now we've extended this now, where you can actually see, we've calculated the decay rates as we did previously. It's about 65 days. That's okay, but not all that better than the Pfizer vaccine. But look what happens when you give it a 3rd boost here. You see, we boosted it one final time, 6 months after the first immunization or longer after the 2nd immunization. And now we improve on the decay rates. Now you're getting to about 134 days or so. And the same thing with the neutralizing titers. Now you might, at face value, you might say, well, 134 days is not all that different from 65 days. But if you work out the math and you work out the numbers, actually, you know, it's actually considerably long. It takes about 2.5 years before you reach a threshold of about 100 in terms of neutralizing antibody. So maybe this is giving us a clue that adjuvants might be helpful. And of course, if you have more durability, then this is good news in terms of the breadth of the response as well. So this is an example of the breadth of the response that we're seeing with the same adjuvant with ASO3. You see, this is what you get for the ancestral strain, and this is what you get against Omicron. So there's about a 5-fold hit or a 6-fold hit. In contrast, with the Pfizer vaccine, we see an 18-fold hit at the corresponding time points, okay? So perhaps adjuvants have a clue. The other lesson is, how do you exploit the synergistic interplay between T cells in tissues and antibodies? We've heard wonderful talks about antibodies and about T cells, but I think the important point to appreciate is to exploit the synergy. So clearly, this vaccine induces decent T cell responses in humans in the blood, and in mice, remarkably high frequency of tetramer-positive T cells in tissues, in the lung, 23%. You see, and this is intracellular cytokine staining, unbelievably high, 65% in the lung. Does it happen in humans? We don't know, but we're collecting bowel samples to analyze this in humans. So why is this important? Well, if you can get a high enough frequency of T cells in tissues, you should exploit the synergy. So in a study that we had done 2 years ago in the context of HIV, we compared vaccination regimens in monkeys involving just a recombinant protein, HIV envelope protein, with an adjuvant alone, or interspersed with a vaccination strategy with viral vectors expressing T cell epitopes, GAG. And to cut a long story short, what we see is that if you had both of these, T cells and antibodies, you have substantially better protection than you would if you just have the antibody alone. In the interest of time, I'm not going to spend too much time on this. And then the last lesson, or the penultimate lesson, is the innate response. I mentioned to you this persistent epigenetic imprinting with adjuvants. Perhaps this could be harnessed in the context of HCV. We had shown this with the H5N1 ASO3 vaccine that I mentioned. And indeed, this epigenetic imprinting that seems to persist for several weeks and sometimes months does seem to have an impact on controlling very unrelated viruses such as Zika and dengue, completely different from the H5N1 vaccine, which when you infect PBMCs from these vaccinated individuals, they seem to be able to control these completely unrelated viruses. So I think that's another lesson. Finally, the most important lesson, I would say, for HCV vaccines, but also for any vaccine development, is the very fact that our capacity now to assess immune responses in humans, such as using systems vaccinology techniques, I believe has really invigorated the ability to rapidly evaluate novel vaccine concepts in humans in what I call phase 0 HCV vaccine trials, early early is the operational word in the vaccine development pipeline. In other words, not wait for 5 years or even 3 years or even 2 years after you have to do laborious mouse work and monkey work. But why not do it in small human phase 1 trial where you can analyze immune responses in the blood, in the draining lymph nodes using fine needle aspirates, even in the liver, to quickly weed out vaccine concepts that clearly are not going to pan out in humans. So with that, I'd like to thank all the people who did the work, Prabhu Arunachalam, Florian Wimmers, Chongfeng Li, Audrey Lee, Lily Gregorian, and this was a wonderful collaboration with all these other labs. Thank you.
Thank you very much, Bali. This was wonderful. Can I maybe start with a question? You know, HCV is a bit a more challenging virus, obviously, than the others. And I'm, as a T-cell person, a bit worried sometimes that we often just look into frequency of cells, but I think that the T-cells quality is what might matter at least as much. And then it becomes really complicated, right? Have you tried to incorporate that kind of in your study to get more of an idea of quality of T-cells, not just frequencies?
Absolutely correct. Thank you. Yes, it's much more, I think it's important to look carefully at the quality of the T cells. As I mentioned in the HIV study, we look carefully at tissue resident memory T cells. I think this is a very important cell population that will no doubt have an important effect in controlling mucosal infections and other tissue infections. And so I completely agree with you. I think in these human studies and in the non-human primate studies, it is absolutely critical to dive in more deeply into what kind of T cells you're inducing.
Thank you. Then I have a question here from Ellie Barnes in Oxford. It says, the mRNA vaccines, both Moderna and Pfizer, have nucleotide modifications that purport to limit interferon innate immune stimulation. This modification seems not to be limiting interferon in your experiments. Can you explain?
Yeah, it's a great question. Wonderful question. They are modified. They have this methylation in the uracil. We make a pseudo-uracil, which is designed to prevent it from activating toll-like receptor 7. They also have purification to get rid of double-stranded RNA structures. And so, as we've seen, TLR7 activation does not seem to occur, which is consistent with the fact that they're modified. However, we do see some level of MDA5 activation, in fact, quite a lot of MDA5 activation, which raises the possibility that even despite the fact that through chromatography that they had tried to exclude double-stranded RNA structures, presumably there are some spontaneous double-stranded RNA structures that form. So although the mRNA modified platforms perhaps do not stimulate as potent an innate response as some adjuvants might do, they nevertheless do stimulate some level of innate responses. So I think it's a question of threshold. Now, the question is, what would have happened had they stimulated even more of an innate response? I think on the one hand, you might think that could be beneficial for the adaptive response because after all, more potent innate response can drive T cells and B cells more keenly. But on the other hand, they may have negative consequences in protein translation, mRNA translation into protein and protein expression. So I think it's a fine balance that the mRNA vaccines occupy, but that balance obviously doesn't seem to cut it to get durable responses.
Yeah, I think the thing the companies were very concerned about was the heterogeneity of the responses that you may get if you didn't make those modifications, which would have all kinds of issues around dosing. And so I think it was partly a kind of strategic decision. So dosing could be, done in a very quantifiable way.
Yeah.
So one question now from Andrea Cox. She says, do you think the waning antibodies seen are due to the close spacing of prime and boost and not an inherent flaw in the mRNA vaccines? Do you see the changes seen in the yellow fever virus also in the J&J vaccine?
Yeah, that's a great question. Yes, the spacing seems quite short, 3 weeks. And of course, the reasons for that were because of the expedited timeline that these vaccines were developed in. Now, what would happen if you increase the spacing? I'm not aware of any systematic study that's aimed at looking at this. It could be that you might improve the durability with increased spacing. But on the other hand, with some of the adjuvant studies we're seeing, even with not exactly the same spacing, but similar spacing, the durability does seem to be better. So, but it's a good point, Andrea. I mean, we can't exclude the fact that it's simply a question of spacing. Yeah. What was the other question?
There are more questions maybe that you could answer in the chat, please, because we need to move on, please. Thank you. So we now come to our next speaker, who does not need much introduction. Michael Houghton is the father of the field. He and his colleagues, Drs. Chu and Kuo, identified the hepatitis C virus in 1989 and also identified quickly antibodies that really had a huge clinical impact very early in the field. When I call him the father of the field, it's not just for his many scientific contributions. I'm old enough now to have attended the HCV meeting almost from the very beginning. And I always was impressed how he used his authority in the field to push us, to motivate us, and especially to remind us that this was not an academic exercise or anything, but that this was really an urgency for many, many people in the world. And he has always made sure that everyone understands that we need to find cures, but also prophylactic vaccines for everyone in the world. So these things need to be soon, and they need to be accessible and affordable for all. And I think he will tell us, after the cure has been accomplished now, more or less, he will tell us now how we can get there for the vaccine side as well. Thank you, Michael Houghton, for being here.
Thank you very much, Georg. Can you see my slides okay?
Not yet. We saw them quickly before.
Oops. Let's try again.
Would you like me to try, Michael?
Yes, please, Lindsay. Thank you. Is it on full screen, Lindsay?
It is, yes.
Okay. Well, thank you, Georg, for your kind comments. Obviously, it would be very nice to develop a vaccine to Hep C, obviously. And I think if we do that, then we can put an end to this epidemic. So thanks to the organizers. So this is my title of my talk. What's the feasibility of delivering a vaccine this decade? Next slide, please, Lindsay. I apologize. Now I can't even see your screen. Are you there, Lindsay?
I am, yes.
I'm sorry, I can't see the slides now. Hang on.
We can see them, but
you can see them, but I can't see them.
But you can see them.
Oh my word. Okay. Thank you, Lindsay. So yeah, it's been interesting. This morning, some fantastic science presented by all the presenters, and there still seems to be a little bit of debate how important antibodies are. So I wanted to start with just some context on this topic. Next slide, please. So, what direct evidence do we have for the protective role of antibodies? To me, the work that Dr. Mühlemann did, published in 2011, has impressed me a lot. So it's using the SCID UPA mouse model developed by my colleagues at University of Alberta, Lorne Terrell and Norm Kniederman and David Mercer, in which SCID mice can be used to colonize human hepatocytes, and then you can infect these mice with Hep B, Hep C, and other pathogens specific for the liver. And so what Philip Mühlemann did was to infect these mice and give irrelevant antibody as a control, or he gave immunoglobulins purified from Harvey Alter's famous patient H. These were antibodies purified from the chronic phase of H's infection. And Jane McKeating and others have shown that in the chronic phase of this patient's infection, there were broad neutralizing antibodies that were present. And so Philip asked the question, what happens if we passively immunize these mice with this purified immunoglobulin and challenge with homologous virus on the left of this panel? And you can see very clearly relative to the control antibody, which was the same just a control antibody isotype, that the HIG protected these animals very well, shown in red. After 2 weeks, there was no infection registered in these animals. Then he went on to challenge with heterologous virus, 4A and 6A. And at week 1, all the control antibodies were infectious with high viral loads. But in the animals that received the IG from H, as you can see, 6 animals were negative for viral RNA. The important thing to remember in this model is that the half-life of human IG is very short. It's only in the region of around a week. So in subsequent weeks, with regard to the challenge with 4A, 2 of the 6 mice were still negative. The other 4 were positive, as you can see. But even so, some of these animals had less viral load than the control antibody mice. And even in week 3, as you can see, there was 1 mouse that was still negative for viremia. And pretty much the same pattern when he challenged these mice with 6A, a very diverse genotype. On week 1, 3 of the 4 mice with the HIG were negative, unlike the 3 control antibodies. So this has always impressed me regarding the preventative role of immunoglobulin, given that the half-life is very low in this animal, very short. Next slide, please. And also I was impressed by this work from Dr. Moran and colleagues in 2012, where he used the chimpanzee model and then passively immunized with the HCV1 monoclonal antibody. The control is shown in green, was not passively immunized. And as you can see in green, the animal quickly became viremic after challenge, experimental challenge. In red was an animal that received the monoclonal antibody at 50 mg per kg. There was obviously viremia, but when he used a higher dose, 250 mg per kg, shown in the animal shown in blue, as you can see, there was no infection. So this is a broadly neutralizing monoclonal antibody, and when it's there, it does its job. It prevents infection. So I think these are a couple of examples. There are others. Manson published data in the mouse using broadly neutralizing mAbs to show that they were able to protect mice as well, the UPA mice. So I think it's important just to remember that there is direct evidence for the protective role of antibodies. Next slide, please. And I guess I've used the chimpanzee model to test HCV vaccine candidates more than anyone else. When I was at Chiron company in California, we used it a lot with different types of vaccine. One of them was a T-cell vaccine. It was a non-structural protein, polyprotein, that we adjuvanted with Iscomatrix, a very powerful adjuvant. And in chimpanzees, we saw a very nice amelioration of acute hepatitis and acute viremia. But when we followed 5 of those animals up, only 1 actually resolved the infection. which was actually a lower rate than the control animals, where 3 out of 5 resolved approximately. 2 out of 5 resolved. So, pretty much it seems to have happened with the Arcturus vaccine. It elicited strong T-cell responses, CD4 and CD8, and yet there was no effect on persistence. And that's exactly what I saw with our version of a T-cell vaccine in chimpanzees many years ago. But when we tested adjuvanted recombinant E1 and E2 heterodimer in the chimp model, we saw a very different picture. And I regret not publishing this data in more detail. We put it into a couple of reviews, as you can see here, 2005 and 2011, with yet more data. But I stand by this data. These animals were very carefully monitored by Amy Weiner, and who was a very thorough scientist. And we're monitoring chronic infection, obviously, by the detection of viremia using PCR. And so we immunized these animals, naive animals, with around 50 micrograms of E1, E2. We did use different adjuvants throughout the study. This was done over several years. We could never, even in industry, we couldn't get enough money to do all the animals in one time. So there were various adjuvants used with the E1, E2. But as you can see, with the homologous challenge animals, 12 of those that were challenged, 5 were completely sterilized. No viremia in blood, PBMCs, or in the liver at any time. And these were the ones that had the highest antibody titers. Now, unfortunately, at that time, this was many years ago, we weren't able to monitor T-cell responses to the vaccine. But at least we did see some sterilization. And in the 7 animals that became acutely infected, when we followed these up, only 2 became persistent. Whereas of 10 unimmunized animals, 7 of them became persistent. And there is a statistically significant difference in the carrier rate. Then, as you can see, when we followed up this work by challenging animals with a heterologous H77 isolate, so our vaccine was made from the HCV1 isolate, which is a 1A, when we challenged with a heterologous 1A in the form of H77. We didn't see sterilization. All 19 vaccinees got acutely infected, but when we followed up, only 3 remained persistently infected, unlike 57% of unimmunized controls. So once again, a statistically significant difference, and put all the data together and it's highly significant. So, unfortunately, we weren't able to do more challenges with different genotypes, but I think, to my knowledge, this is the only Hep C vaccine candidate that has been shown to reduce the incidence of chronicity and viral persistence. So that's why I've stuck with E1, E2 for so long. Next slide, Lindsay, please. Thank you. So on the basis of this chimp data, the NIAID co-funded a phase 1 clinical trial. The vaccine was obviously made at Chiron. And we used MF59 adjuvant and oil in water emulsion. And we immunized individuals with different doses, either 4 micrograms, 20 or 100 micrograms, given 4 times. And to summarize, we got a satisfactory safety and tolerability profile. We saw anti-E1, E2 ELISA titers in the same range as in the protected animals. And this is work by Sharon Fry, who conducted the phase 1 trial in St. Louis, and Ranjit Ray did some neutralizing antibody assays at that time. Most vaccinees elicited significant titers of neutralizing antibodies to the heterologous 1A pseudoparticle. We saw very strong lymphoproliferative responses to E1, E2. Mainly those were CD4 T helper responses. And importantly, E1E2 is a very difficult protein to express and purify, as probably some of you know full well. The good thing about this trial is basically 4 micrograms is pretty much optimal for antibodies and for T-cell responses, lymphoproliferative responses. So that's very important, because although E1E2 is difficult to manufacture, a moderate scale up by industry standards would be something like 1,000 liter cumulative bioreactor volume. And at the yields we've been getting, we have two methods to purify it, but using the most efficient method, this level of bioreactor would yield enough antigen for somewhere between 500,000 to 1 million recipients who would receive 3 doses each of 4 micrograms. So, yes, it's relatively low yielding in a industrial vaccine world, but it still can provide enough vaccine, I believe, especially for the high-risk groups who are the people who inject drugs. Next slide, please. Yeah, so a lot of great work this morning was presented on the issue of cross-neutralization. And I certainly accept the points that Justin and Manson were making. I sort of have a little bit of a different perspective. And I guess my perspective is that this wild type E1E2 from a single strain doesn't have a bad cross-neutralizing profile. And I would make 2 other points. One is that traditionally the vaccine world, the corporate world, deals with heterogeneity by combination vaccines. For example, pneumococcus vaccines have now, I believe, 30 antigens from 30 different strains. In the viral world, we've known for decades that poliovirus vaccine is a trivalent vaccine comprising the 3 serotypes. Papillomavirus vaccines comprise 4 different serotypes. So I think the work that was being reported this morning is fantastic and really, really excellent. And it obviously could yield very good improvements to our vaccine antigens. But let us not forget that the traditional way to deal with heterogeneity in commercial vaccines is combinations. So if we use the combination, for example, of genotype 1 and 2 and 3, I take Justin's point, we won't neutralize all the serotypes within those major genotypes, but we will get a pretty good coverage. And the second point is that nobody knows what level of neutralizing antibodies we need in order to be protective. I think that came over loud and clear this morning. And so let us not talk ourselves out of testing available vaccines now for efficacy. I think that's my message. And very quickly, I'm running out of time as usual. This is old work from Jane McKeating's lab in guinea pigs. And what she's showing is cross-neutralization of 1A in black. These are different guinea pigs. Cross-neutralization of 1A in black, 2 1B strains in the stippled and in the clear bars. And even against 2A, the J6 strain, she saw significant but reduced cross-neutralization. And next slide, please. We gave our vaccinated chimpanzees to Bob Purcell, and in his lab, J.C. Mounier and Jens assayed for neutralizing antibodies. They did CC as well as PP. We gave them 4 chimpanzees. And so you're looking at the neutralization from those anti-sera against all the major genotypes, 1 through 6. And what they found was, pretty darn good cross-neutralization for most of them. Genotypes 2 and 3 were less well neutralized, but they're still partially neutralized. And we don't know what levels of neutralizing antibodies are needed for protection. So I certainly agree that we should try and optimize cross-neutralizing antibody profiles, but let us not forget, these might be sufficient, especially as we are doing, if we are going to make combination vaccines of genotypes 1, 2, and 3 to test in humans. Next slide, please. And then John Law, my colleague at the U of A, University of Alberta, as you know, he published some data with the human volunteers that got MF59 adjuvanted E1E2, and we're only showing 3 of the volunteers here, but we saw a profile somewhat similar to what Bob Purcell found with our chimps, a broad cross-neutralizing profile. Given that this is just from a single 1A strain, this is pretty remarkable, I thought, and still do. Genotypes 2 and 3, once again, were not as well neutralized in the human volunteers as the chimps, but historically, we've solved that issue by adding making combination vaccines. Next slide, please. And then one of our students, one of our excellent students, we had 2 excellent students, Janelle and Jason, and Janelle published a paper a couple of years ago showing in goats, we immunized goats to get a lot of anti-sera so we could do more. We didn't want to use the precious human anti-sera. But Janelle showed in the goat model, at least, she was able to get very good neutralization of JFH1. J6 was not well neutralized, but JFH1, this certainly confirms what Justin was saying this morning, that neutralization is not necessarily genotype dependent or subgenotype dependent, but at least it shows this E1E2 can neutralize a lot of different genotypes and a lot of different subtypes. Next slide, please. Next one, please. Yes, we had some discussion yesterday that Stephen rose in discussion. Obviously, very nice work from Jens and other people showing that if you remove the HVR1 region at the N-terminus of E2, the virus becomes very susceptible to neutralizing antibodies. And so the hope was if we remove that from our E1E2 molecule, perhaps we can increase antigenicity. But next slide, please. In terms of reactivity with bNAb monoclonals, we saw a reduction in activity to the top 3 antibodies. These are, I believe, broadly neutralizing monoclonal antibodies, including AP33. The deletion actually reduced immunoreactivity, but a lot of the other good bNAb monoclonals, 8426, 3B, 4A, 5A, we saw no difference. But what that, next slide, please, what that translated to in terms of antigenicity is shown on this slide. We immunized guinea pigs, and then we tested for cross-neutralize. Well, first of all, we tested for neutralization of homologous virus, H77, and you can see that the deleted E1E2 was not as good at neutralizing homologous virus as the wild type, undeleted. On the right-hand side, you see that they were pretty similar in terms of neutralizing HK6A CC virus. So overall, we didn't really see an improvement by deleting HVR1. Next slide. So we've gone ahead and we've made E1E2 from strain H under GMP. I believe when it's combined with perhaps genotype 3 E1E2 and perhaps genotype 2 E1E2, we will get broad neutralizing activity. It may not be perfect. I agree with Justin and Manson, but it may still be effective. And anyway, I'm showing you here the GMP produced E1E2. In track 1 on the left is a silver stain, and that's showing E2 alone that was secreted. And it's the ectodomain from 384 to 688 that was purified. Track 2 is one of our GMP batches. And you can see E2 and E1 fairly clearly. And then in track 3, we've got sort of the old Chiron vaccine, E1E2, that's been purified with GNA lectin. Once again, you can see the E2 and the E1 pretty clearly. And then track 4 is another batch of GMP produced material from University of Alberta. And then the Westerns are shown on the right-hand side. The E1 is obviously lighting up very well as the E2. So we've got pretty good GMP material now. Next slide, please, Lindsay. What we've done with it is used Manson's and Stevens' very illuminating monoclonal antibodies. Most of these are broadly neutralizing monoclonals. They neutralize most of the genotypes quite effectively. And this is interesting because it shows that the E1E2 batches shown in red and blue are reacting with these monoclonals, binding to these monoclonals better than E2 alone. You can see that with the top 3 antibodies, AR1 through 3A. And then on the bottom are Manson's AR4A and AR5A monoclonals, which depend on both E1 and E2 for binding activity. And you can see we get very good activity of our 2 GMP batches, equal in binding to the, if you like, the Chiron-like E1E2 that we tested in phase 1. And importantly, there's no binding of these important monoclonal antibodies to the E2 alone antigen. And then the next slide are some of the results with Stephen Fong's antibodies. His 205 and 210, shown on the right-hand side, are binding in the same region as Manson's AR4 and AR5 regions. And we saw the same picture. The E1E2 GMP material binds very well, but the E2 does not. And on the left, one of Stephen's antibodies, HC84, which is binding in the AR3 region, as defined by Manson, is binding to the E2, but not as well as against the E1E2. So I think this is important because, in my opinion, E1E2 is the best antigen to use to elicit cross-neutralizing antibodies. Next slide, please. As I'm saying here. And of course, with the fantastic work done by Oxford and AstraZeneca and BioNTech and Moderna, we now have the option of delivering these antigens through mRNA or through viral vectors. Next slide, please. It turns out that a group at Novartis back in 2015, this was the group of Andrew Giel and Dr. Brito, published actually 7 years ago, surprisingly, in the results of immunizing mice with 10 micrograms of self-amplifying RNA encoding E1E2 heterodimer. And they compared it with immunization of mice with 1 microgram of my adjuvanted E1E2 protein that I produced when I was at Novartis and Chiron. And well, it's an initial experiment, but I think what it shows here is the inhibition of neutralization. You can see that the protein, the 1 microgram of protein, seems better than the 10 micrograms of self-amplifying RNA. But given that this is the first attempt, I think it's telling us that we can deliver E1E2 through either self-amplifying RNA or through mRNA, and I'm sure through adeno-based vectors as well. Next slide, please. So my feeling is we have many options now available for developing a Hep C vaccine. I am very optimistic. We have the protein-based vaccine. I personally think it should be E1E2 heterodimer. It can be enhanced the old-fashioned way by making a combination of antigens from genotypes 1, 2, and 3. I think we will have a pretty good cross-neutralizing profile if we do that. Justin will argue quite rightly that it can be improved, but I think it's not bad. We can scale this protein up. And of course, if you deliver it by mRNA and vectors like adeno, it should be a relatively easy scale up. We are, like many groups that I've heard at this meeting, we're in addition planning to dial in T-cell responses. What we're doing is a bit different from Okiris, for example. We're focusing on highly conserved epitopes. And to do that, we're incorporating synthetic peptides to highly conserved T helper and CD8 epitopes. And we're using a cocktail so that we can cover many MHC alleles so that we can hopefully induce T cells, CD4 and CD8s in most of the human population. We need to use a good adjuvant. My colleague, Dr. Amir Landi, has done a lot of work in the mouse looking at different adjuvants. We're impressed with the data from Bali using the TOL7/8 agonist adjuvants. We've been looking more at TOL4 agonist adjuvants, and we've been impressed, certainly in mice, with the strong responses, both CD4 and CD8 that Amir's been getting with E1, E2. And I'm actually encouraged by a paper from Adam Penn Nicholson in Vaccine 215. They had a fusion protein made from TB, tuberculosis, and they used AS01, this GSK adjuvant, and they showed in humans that they could get very strong CD4s and CD8 responses to that antigen. And as I said, we've got the mRNA and the viral vectors now to deliver these antigens. Next slide, please. So my belief is that we can roll out an effective Hep C vaccine, at least to the people that need it the most, who are the people who inject drugs, this decade. I believe that's very feasible. I also believe, as you've probably gathered from my talk so far, that I believe the wild type antigens that we're using are more than worthy to continue clinical development. I think there will be multiple phase 1 trials, I'm sure, reported over the next 1 or 2 years from many groups. And if we get good, reasonable cross-neutralizing antibody profiles, if we get cross-reactive CD4 and CD8 T-cell responses, and if the CD8 responses are presented by multiple MHC alleles, then I think we should proceed to efficacy testing. Now, Jake Liang organized a very nice meeting last year at the NIAID to discuss human challenges, and I think this is a good way to go. We are talking to regulators in Health Canada right now about how to do these trials, and I do think, I agree with Jake and colleagues that this is a good way forward. So I expect, hopefully, to be seeing some efficacy reads out in humans in 24 or 25. And I think if we can show that volunteers that are vaccinated and then challenged actually go on to develop a very low chronicity rate, that compared with all the studies, historical studies, where we know what the chronicity rates are for lots of different populations around the world, and they're very high, as we all know, I believe that should allow us to start rolling the vaccine out in a staged, sequential way to people who inject drugs. And then we can follow up larger numbers of those individuals to confirm that the chronicity rate is a very low level. And then after that, I'm sure the brilliant work from Manson and Justin and everyone at this fantastic conference has talked about, we will, I'm sure, get a superior vaccine emerging over the next few years. And then we can start dialing that in, of course. So next slide, please. I think it's my last. Yep, just like to thank all the many colleagues I've had at the U of A, the research team of John, Mike, Amir, Judica, Darren, Jason, and Janelle. We have a large GMP team that are really doing heroic work. E1E2 is not an easy protein to make. My dear friend and collaborator, Lorne Terrell, has supported us all the way. Would like to acknowledge that this work started many years ago at Chiron with my co-discoverers of the virus, Quilin Chu, George Kuo, and then also involved the vaccine work, also involved Bob Ralston and Richard Spady. Manson's and Stevens antibodies have been very, very important in our interrogation of E1E2. Thanks to them for providing those antibodies at all times. And I'm really enjoying the U19 team that I'm on with Stephen, Bally, Chris, Richard, and Francois, and my colleagues at the U of A. So thank you very much, and happy to take any questions. And I guess we'll also have a discussion period coming up.
Yeah, thank you very much, Mike. This was great for giving a bit of a different perspective. And I don't have a question right now online, but it seems to me, that the really critical thing to get this all happening is really to get the controlled infection models going, because, then we can quickly test whether your kind of more, let's call it classical approach works. If so, great. If not, we're fine. The issue will obviously be, do we get it? Because I think if we do not make this happen, then we are in trouble. The last trial took forever, was incredibly costly. And I mean, you cannot always wait very much 5 or 7 years. Andrea can tell us how long it took from conception to conclusion, to know that, okay, this was not the right idea. Let's move on to the next. So that seems to be maybe one of the key things we should have in the discussion afterwards in the round table, because that completely changes our planning. If we could, and Bali said also, right, we should test quickly. And I completely agree, even from failure we learn, but we can only learn if we can do them in different approaches and relatively quickly. And well designed, obviously.
I agree.
So since no one else came forward, then maybe we can go straight into our discussion panel. Obviously, Mike will be a part of it. So Stephen will lead this discussion. Stephen Fong from Stanford, Chris Walker from Nationwide Children's will be part of the panel, David Thomas from Hopkins, and Marion Major from the FDA. But I think the other panelists also might chime in potentially. So Stephen.
Yeah, thank you. The terrific presentations for the past 2 days have highlighted really quite promising approaches for vaccine development. I believe that there is now a consensus that to succeed, vaccines should elicit broadly neutralizing antibodies and T-cell responses, recognizing the diversity of HCV isolates. We definitely need more powerful approaches to assess immunity that could provide new insights to determine the quality of vaccine responses in preclinical and clinical HCV vaccine studies. However, there are notable challenges in assessing HCV efficacy in humans. There are fundamental questions which are highlighted here that really we should address, which is really the focus of the panel discussion. The 3 questions are: how do we design a universal vaccine? How to recognize a potentially useful vaccine in the absence of a preclinical challenge model? And 3, when to advance an HCV vaccine to human trials. Next slide. In the first question, is it possible to design a universal vaccine? I like to call on the B cell side, I like to call on Manson, Justin, and Jans in that order to address the first question. Is it, can we design a single vaccine sequences that elicit neutralizing antibodies against the diversity of isolates among each and all genotypes? And clearly, Justin has presented very elegantly that we need to concern about serotype or phenotypes. And then the second question is, if the answer is no, what type of a multivalent vaccines would be needed? And Mike has really addressed that in his talk. So Manson?
Yeah, I think for science, everything's possible. I think we have to be optimistic about that. But I feel one thing important is every time we do some immunization, we need to find out what's the outcome and results. And also, I think Stephen and Justin, we all agree we have to standardize some of the assays so that we can generate compatible results and keep improving based on that. I think the field is going to a good direction in there. I think Justin developing this panel of virus. I'm piggyback developing the bunch of antigens that can be used in immunoassays and standardize the method so everyone uses the same method so that we can improve upon that. I think is really the key. So I'm very optimistic. And in terms of whether a single sequence or mix and match type of approach, I think we've yet to see, but at least on my end, like noticing this so-called the multi-donor class of polyneutralizing antibodies, to me is pretty groundbreaking in the HCV field. And we can try, one of the major approaches in my lab is try to target this type of polyneutralizing antibodies, see whether we can improve their yield and production, and also whether we can find a way to specifically elicit this polyneutralizing antibody. So currently, of course, we are doing this single sequence approach, and maybe we need to mix and match, but those can be tested at least, like for example, in the monkey model. And so I'm very optimistic about it, yeah.
Mm-hmm. Justin?
Yeah, I agree with Manson, and I mean, I would say we don't know the answer whether a single strain or isolate is going to be adequate to induce broadly neutralizing antibodies. I really actually agree with Michael that it's the single, HCV1 or H77 isolate does surprisingly well at inducing cross-neutralizing antibodies. We do see that people who are repeatedly reinfected with different strains of virus do develop more broadly neutralizing antibodies than people who are infected chronically with a single strain. So I think that it's likely that a multi-isolate or mosaic antigen might ultimately be better. But I agree with Michael that we actually don't know, and it's still possible that a single isolate would be adequate.
Yeah, I think, thanks, Justin. Could I comment, Stephen?
Absolutely.
Yeah, thanks, Justin and Manson. Congratulations, your work is beautiful. Really enjoyed listening to you. It's really, given that it's E1E2 and it's a hell of a protein to make, and congratulations to Thomas first on getting some progress on the secreted antigen, E1E2. But it's relatively easy to make a combination of E1E2s from different genotypes. I think we know how to do that now. I agree. I think using your panel of 15 key pseudoparticles, let's try and show what is the best combination for neutralizing those. We have to bear in mind, of course, that if some are less well neutralized, does that mean it's going to be ineffective against those viruses? So maybe I would imagine, based on your talk this morning, that you will not be able to predict, perhaps, if a low response to some of your 15 pseudoparticles means they're not going to be effective. Is that correct? Would that be your interpretation?
Well, I mean, one thing I can say is that we've tested sera from a bunch of humans who spontaneously cleared HCV, and not a single serum sample neutralizes all 15 of those pseudoparticles. So I do think that, requiring a vaccine to neutralize all 15 is probably too high of a bar. I think the use of the panel is just so that we can compare apples to apples across different strategies. And whether it's, 10 out of 15 neutralized or 12 out of 15 neutralized, at least we'll be able to see, which vaccine seems most promising.
Mm-hmm. And if I may, Stephen, shut me up if I'm talking too much, but I'm really would like to ask Justin and Manson, I presume you would want any envelope glycoprotein antigen to be used as a vaccine, to have these neutralizing antibody epitopes that require both E1 and E2. Do you agree that that is a criteria for advancing an envelope glycoprotein into clinical trials or not?
The short answer is yes, I agree. We've looked at the antibody types induced in people who clear versus persist, and we see AR4A-like antibodies in people who clear. So it does seem important to have antibodies that target this E1, E2 complex as opposed to E2 alone. I think it's still possible that if you got super potent antibodies targeting the CD81 binding site, which is just an E2, maybe that would be adequate. But I agree. I would advocate for E1, E2 as an antigen.
Yeah, I think your point really, we need to test out all this possibility. I mean, so far I feel the pipeline is still relatively narrow, and also each time there's a candidate, I think finding out what sort of immune response is important. And we still do not know whether you need very diverse cross-neutralizing antibodies for protection or just one or two. But some work done previously in Charlie's and Alex, they showed that, of course, you have multiple polyneutralizing antibodies with over better protection than one. So if, I mean, a lot of the antibodies we found is to the E2 neutralizing phase, the receptor binding sites. And the AR4, which is at the interface, mostly on E2, and it's very broad. And that one, I think now, when the structural information come, I think that's another area that I think should have a lot of intensive effort to see whether we can engineer that. Of course, naturally, with the E1, E2 complex you make, and that produce very, very good, preserve the AR4 epitopes very well, I mean. And I think Thomas, maybe you can comment a bit on that, Thomas, first. Like, I think your earlier version of the soluble E1, E2, when with the LC dimerization domain, the AR4 was there, but it wasn't as great as later on you improve further with the SC dimerization domain. So their way or their to further improvement, and once the thing's been improved, you really can increase and see the binding of AR4A much better, which is something that we notice, like when we make E1, E2, and then quite often the AR4A doesn't bind very well. It's indicative, it's a mixture of folded versus not natively folded proteins on there. Yeah.
Mm-hmm. Thank you.
Before Jan's comment, I'd like to raise a different point. Perhaps one of the difficulties of inducing BNAbs is that we know for some of these, at least, they are rather cryptic. Probably the best example was actually one of the first BNAbs that have been characterized by Arvind Patel and Jonathan Ball and their colleagues, and that is the AP33 epitope, or the AS412, which is the newer designation. And that we know that based on their work, they have shown that this sequence just downstream of H3L1 is highly, highly conserved among all isolates, not just within a particular genotype, but really among all isolates. But yet at the same time, only a tiny fraction of individuals that are naturally infected actually produce antibodies to that sequence. So the implication of that is that these that BNAb epitopes can be cryptic. And more recently, Jans have really described some really very important work. And I would ask Jans to comment in terms of, given the data that's out there, the findings that are there, what are, is it possible for us to stabilize or to better expose some of these BNAP epitopes in such a way that we actually can get higher affinity or broader breadth of these types of antibodies?
Thanks, Stephen. I actually don't know if I'm the right one to answer that, but I would just say how fantastic it is to feel this excitement there is for a vaccine. Just 4 years ago, many of us felt the necessity to get together to write a consensus paper on trying to convince everybody that it was important to develop a vaccine against HCV. So this development is really great. Also, as being the one that originally were the first to publish the 6 major genotypes of HCV, I'm of course a big proponent of the importance of these genotypes, both for diagnostics, for treatment outcome, and also for vaccine development. But still, at this point, I'm a little bit in doubt. The question was original question was whether we can get away with just one strain to vaccinate with, like Michael has done with the HCV1, and seen actually quite broad protection. And Judith Godwine and her group in our COHEP program recently published a paper on a whole virus inactivated vaccine with just one strain. And there she saw excellent cross neutralization against, in that case, we tested against 7 different genotypes. And so that shows that just one antigen can at least give that kind. And I think that is also what we've seen in chronically infected patients early on. So there is that kind of cross neutralizing antibodies against the different genotypes. And I agree, neutralization is not determined by specific genotypes. It can very well, 2 different strains, closely related, can have very different neutralization profiles. So that's one observation. The other observation, of course, is that there is these things, as we also talked about yesterday, that I think Yannick Prantø also in our program several years ago showed that if you take the HVR1 out and then look at how these different strains are neutralized, the big difference between them completely disappears. So if the HVR1 sequence is gone, they all neutralize the same. So that kind of means there must be some epitopes in those sequences that are highly conserved. So if we could get at those in some ways, then that would certainly be helpful. And then finally, I think when it comes, I guess this was just with the antibodies, but I did want to make the comment also with the T cells. I think it was very apparent from early chimp studies, including one that I did myself, that if you have an animal that's infected with one strain, yes, you get, and we saw data also from Robert Lanford and Stuart Ray yesterday, if you're infected with one strain, you can get infected short-lived with other strains, but you many times will clear. And we did that with different genotypes also, and so did others. And what we found was that those animals had absolutely no neutralizing antibodies, yet they cleared one infection after the other, showing that the T cells alone could clear those infections again and again. So if we can combine those 2 components, the broadly neutralizing antibodies and those effective T cells, I agree with Michael that this is possible. Then coming back to your real question there, Stephen, I really at this point I'm not sure how we can. I think there's so much great work going on now on how to modify these envelope proteins, and I just think we need to see where it's all taking us and be very open to testing. And I really agree with that. We should not put any limitation. We should not say that because designer-based, that's wonderful, we can change the antigens, but I also agree with Michael, we should go ahead and test the authentic antigens in all kinds of formats. And then finally, I would just caution that if we have these, and I think it's great that these panels have been developed, both the CC panel from Pietzman's lab and the PP panel from Justin Bailey's lab, and they're really great tools to have. They're based on a lot of work from before. And I think what we just all have to remember is how important that when we use them, that we make sure, and that's especially true for the CC, that the envelopes don't change, because just one nucleotide change can give a 3-log difference in neutralization. So it's very important that we have the viruses that we use for neutralization assays under control.
Continuing on the T cell side, Chris, Arash, Andrea, Giora, would you like to comment?
It's Chris here, Stephen. Sure, thanks. I think that for T cells, we also don't know if a single sequence will suffice. As Jens just pointed out, a single infection with one genotype of virus can provide apparent protection from persistence with a different genotype. Whether we can achieve that with vaccinations, not known. I think it depends on how you approach the problem. There are clearly some approaches that involve identification of conserved sequences or epitopes across genotypes, and that those can be used. And I know that Mike Houghton and his colleagues at the University of Alberta and Ellie Barnes and her colleagues at Oxford are looking at those types of approaches. The other involve the use of consensus sequences, ancestral sequences, as championed by Stuart Ray for quite some time, and now epigraph or mosaic sequences from the Los Alamos group and our colleagues. It seems that at least with the mosaic type approach, a polyvalent vaccine will probably be required. It may be that will be easier to achieve with something like an RNA vaccine, which appears to give good T cell immunity, than it would be with construction of different viral vectors, although that's arguable. So a lot will depend on the approach, the vector system, how easy it is to make polyvalent. The one thing I will indicate, and I noticed there was a question in chat, it will be necessary to use the non-structural sequences as they are the dominant targets of the T cell response in HCV. So I guess that would be my comment.
Arash.
Yeah, thank you, Stephen. I think I'll follow up with Chris's point, and I guess, you know, one important thing for me is, and I think Chris really is working on this topic as well, because having access to the actual site of viral replication in the liver these days is very difficult. So for me, the question is actually how many epitopes are being targeted by T cells? Is it, you know, few at one, few, or many? And then how many are really required in cases in which you know, either in humans or animals have resolved versus gone persistent? So I think for me, I still the question of whether we need to target, you know, multiple epitopes, although that, you know, what Chris Beasley showed that, you know, years ago, and that, you know, at least the ones that resolved, they seem to be targeting multiple epitopes. But in actuality, in the liver, in the site of active viral replication, how many epitopes are being targeted and expanded in the liver, I think is a question that is of interest to me.
Andrea?
So I actually think that there is some evidence from COVID vaccination that's actually helpful here. I actually would say that the mRNA vaccines, as I think everyone knows, the platform was not initiated at the onset of the COVID pandemic. And in fact, Justin has been pursuing that in combination with the team at Penn, Drew Weissman and others, for HCV vaccines as well. But one thing that seemed apparent from the influenza vaccines generated with mRNA is that they don't induce CD8 T cell responses as robustly as vectored vaccines, for example. So I'm not sure that that is going to be the ideal platform for induction of CD8s. The other thing that I think is informative from the platform from the COVID-19 vaccine efforts is a recent study by Bob Cedar demonstrating that the ancestral sequence which was really, the parent vaccine sequence essentially because it was made early in the pandemic, actually generated more cross-reactive neutralizing antibody responses against Omicron than an Omicron specific vaccine did. And so I think that in the case of very diverse viruses, potentially from an antibody and a T cell standpoint, there is an advantage to using ancestral or consensus sequence. And that may actually be better than conserved epitopes because some of these epitopes it is at least possible, as Chris, Georg, Barbara Rehrman, and others have shown, it is possible to escape T cell responses, including very commonly recognized T cell epitopes. So I think it's complex, but I do think we should learn from what's going on in development of other vaccines, influenza, SARS-CoV-2 vaccines, to try and inform some decisions. And I also agree with Georg. I think the one thing I will point out about the use of CHIM, because I will say, having conducted the first efficacy trial for a prophylactic HC vaccine, it really led to people becoming very discouraged. And I do think that every failed vaccine trial has the potential to worsen that. So I want to be thoughtful about bringing vaccines into at-risk populations where we think we may really actually provide benefit.
Kira, any comments, additional comments?
I want to add a little bit in a different direction from what was said up, because I think the real problem for the T cells here in HCV is that just like in HIV, we have a really challenging virus and we do not know what protective immunity on the T cell side is. Like, and maybe same for the antibodies. We really don't know. It could be, we think good breadth and good magnitude is important, but it seems certainly not sufficient. I mean, Andrea's studies in her cohort and some of ours show we have patients with incredible T cell responses that become chronic. So quality is maybe just as important, or characteristics of, and what the vaccine can induce. And since we don't have the target of what we need, it makes it really problematic. So that's why I think CHIM is so important, because the more things we can try, and hopefully the more we can learn, even if it doesn't work without having spent many, many years. And the other thing is, obviously, we always talk about CD8 T cells, and in HCV, it's pretty clear that without CD4 T cells, we have a problem for both the antibodies, most likely, and certainly for the T cells. And for them, our knowledge is on one hand even more clear, and that is without CD4 help that's sustained, there's no clearance, but at the same time, we know much less about these cells. So I think the best we can do is hopefully get these controlled infection scenarios started, plan them so well that we really get as much information about immunology out as we can, and then make better informed decisions if it doesn't work as we think it hopefully does.
Thank you. I think we should move on to the remaining questions on this portion anyway, and probably tackle both question 2 and 3 at the same time. And that is whether there's a common definition of a broad response, whether it's on the B cell side and the T cell side. Justin, if you could provide some additional comments, because you've shown in terms of like the importance of the diversity of panels in genotype 1, do you think that we need to do more for the other genotypes?
Yeah, I mean, I'm like a dog with a bone on this question. I think there is no standard definition of a broadly neutralizing antibody in our field, and that is a problem, full stop. And I think that we should work together, all of us, to define a broadly neutralizing antibody, either in serum or a monoclonal, based upon a standardized panel. And a lot of us already work together, Steve and Manson, lots of others, to try to come up with this panel that I talked about today. So I hope that that can move us in the right direction in coming up with a standardized definition of a broadly neutralizing antibody. And I don't know the answer to the T cell, of course. I would defer to someone else on that question.
On the T cell side, who would like to take the lead on that? A common definition. Chris?
I think on the T cell side, the question of what a protective response looks like is still up in the air, especially on the CD4 side. Tobias Beutler and others have showed that follicular helpers, T helper cells, are an important component of the response. In immunogenicity studies, we tend to still look at Th1 type cytokines. It really seems like we may need to broaden the analysis, perhaps using the systems biology approach that Bally described. But I think, as Georg pointed out, the quality of the response is important. And as with antibodies, we don't have a standard definition of quality at this point, and what readouts we should be using. And I think it would be important to come to agreement on that.
Yeah, it looks like we're really running short of time. So what I'd like to do is move on to the next slide. Lindsay? Right. The next set of questions are really how to recognize a potentially useful vaccine in the absence of a preclinical challenge model. Bali has really elegantly talked about the lessons learned from the SARS-CoV-2. David, could you provide additional comments in terms of what you feel are the lessons learned, and what more we could glean from those studies?
Was that directed to me, Steve?
Yeah, you and also David Thomas.
Oh, okay. Yeah, I'm not sure I can give a good response to these questions. I mean, we've already discussed, what is a successful immune response at the B and T cell levels. And, I think all we can do is kind of be practical, right? If we're looking at different vaccines, we would like them to neutralize many of these nice panels that Justin and Thomas Pichman have put together, right? If we have one that is very poor at neutralizing most of those, then we wouldn't be too enthusiastic about putting that forward. And the T cell, I must admit, I talked to some GlaxoSmithKline vaccine people a few years ago, and I told them the chimp data we had with E1, E2, and they seemed to be impressed. And I told them, well, we're also going to dial in T cell responses. And they said, well, why would you do that? Why don't you just go with E1, E2? And, obviously at the time I was influenced very much by the great T cell immunology that's been done in the field. And I thought, no, we got to have T cell responses. And I still feel that, but I guess I am a little bit, I'm a little bit perturbed about the Akira's trial not working. And I guess I've been influenced by the studies I briefly mentioned in my talk this morning, that when we vaccinated 5 animals with our T cell vaccine, a big polyprotein, very similar to what Akira's expressed in from their vectors, we expressed a big protein in yeast, 2,000 amino acids. We combine it with a really good ISCA matrix adjuvant. Using Chris's great methods, we show that prior to challenge of those animals, we had multifunctional CD4 positive T cells specific for the virus in the liver. And we had multifunctional CD8 positive T cells in the liver prior to challenge. And yet when we challenged, everything went well for the first few months. Very low viremia, very low acute hepatitis. But boy, when we followed up those 5 animals, it took almost 2 years for one of them to resolve the viremia. The other 4 were persistently infected. And so I guess I'm just kind of a stream of consciousness here, but I do sometimes, I guess I feel it's very important, as Andrea's doing, trying to find out why that trial did not work. And I've been influenced by your work, Chris, that we know that CD8 responses that the virus can evade those. And you know, I think the quicker we find out if that's what was going on, then it's really going to help our field. And yeah, I'm continuing to dial in T-cell responses with my colleagues. We think, I'm kind of assuming that the reason the Icarus trial failed is because the viruses evaded the CD8 response. And that is why our strategy is to really focus on small, highly conserved epitopes, T-cell epitopes, and use really good adjuvants to prime those in people. And as I think I mentioned, there's a nice paper with TB antigens using AS01 that did prime very good CD4s, very good CD8s in humans. So I'm not saying we're going ahead with AS01 because there's some other issues with that it's a very reactogenic adjuvant. It's used in the shingles vaccine. A lot of people get quite sick from that shingles shot, and it's only 2 shots. Well, I believe, I guess I've been influenced by Hep B. I believe we need 3 shots for Hep C, and I'm not so sure, especially people who inject drugs, whether they're going to come back to get all 3 shots if we use AS01. So whether we should use the one that the adjuvant that Bali's been showing nice data for, the Tol7, Tol8 agonist, or whether we should use the Tol4 agonist needs to be decided.
Can I maybe answer to Mike quickly? Because I think even, for example, if we see now that there were escape mutations in the trial, you know, we need to be very careful. We need to find out, was it consequence of persistence or the cause, or was it the consequence of having T cells that did not have the quality, or did we not have enough CD4 help? Or so there are just a lot of opportunities. It is complex, and that's why I really hope that we can see a number of different controlled exposure trials in the next few years, that we can sort this out a bit, because there's nothing better than, I mean, mouse data and stuff are not helping us, obviously. We need to see what certain things do and what happens to certain immune responses when a human gets exposed. And if we can observe it in real time, because that's the other thing, obviously, compared to the regular trial, apart from being much less costly and shorter, et cetera, is that we can observe when the inoculum goes in real time what happens. We can do this work that Bali has pioneered, you know, these kind of things. We can look at everything and know know the sequence of events. And I think then we might be finally able to learn, you know, what is needed and where exactly does the defense get cracks. And I think that will really be very informative. And so I just think it's too early to really, you know, go on just put on the money on one horse.
Mm-hmm. Yeah, good points.
David, Bali, do you have additional comments for the few minutes that we have for this set of questions?
No, these are excellent comments, and I very much agree. And you know, sort of highlighted how I thought what the lessons were at the end of my talk. So
I think we need to move on to the last set of questions. And that is when to advance a HCV vaccine to human trials. And these are the questions: whether requirements for vaccines to enter phase 1. And second question, what population should be included in the phase 1? Should we go beyond people who inject drugs? And if we think that would be the case, what would be that population? And lastly, what are the metrics to advance an effective vaccine candidate? Marian, would you like to take the lead on these questions?
Yeah, sure. Thanks. So the first question, and I think that there have been a lot of really important comments about moving into human phase 1 trials. And I really like the last point that Bali made in his presentation of basically looking at them as like phase 0. And all of these discussions about the human challenge model, I think they're important, but that's a massive amount of discussion that still needs to go on. But you don't have to wait for something like that to move something into a phase 1. And as so many people have said, and I think Mike has said, you don't know what's going to happen in terms of human immune responses unless you immunize humans. There isn't really a model that we can use to predict. And you don't need a huge amount of information in terms of what the FDA might require in order to go into a phase 1. We would want to see some reassurance of safety and immunogenicity, but a phase 1 trial is to test safety and immunogenicity in humans. We're not asking to be convinced that this is going to be protective. We're not asking that this is going to be moved into a phase 3 trial. That's for vaccine developers to decide if this is what they want to do. And so these things, it doesn't have to be sequential. And I feel as if sometimes we're getting a little bit too sequential with this. We want to sort out this problem before we move to the next problem. And there's so much information that can be gained, and you don't have to have the perfect candidate for a phase 1. And it's not uncommon for sponsors to come in with different candidates or variants of one candidate to see which one they want to take in to the next phase. So I really would like to see people thinking much more about what to take into a phase 1 trial. And finally, just to encourage people to come forward with pre-INDs. I don't know if people are familiar with pre-IND meetings, which would come before an IND application, but you can ask FDA a lot of these questions that you may not be sure of. What do you need? What do you need about immune responses? And the last point on that last slide about standardized assays, which is important, but again, they don't have to be established before you go into a phase 1. So I really think I'd encourage people to think much more openly about trying vaccines in humans at this point. Thanks.
Andrea, David, would you like to comment on this topic?
Dave, you want to go first and then I'll comment?
No, go ahead, Andrea. I think it's, you did a study. I think you have the right to speak.
Well, and I think we all have a lot of collective experience. I agree completely with what Marion said. I mean, I think part of, and what Michael was saying, that I think, you know, it would be helpful to do head-to-head comparisons of things we've theorized might be advantageous. And we're trying to do some of this work in animal models, but we know the inherent flaws to that. So I do think that a head-to-head comparison of various strategies for inducing neutralizing antibodies, I completely agree with the need for consensus on what is defined as a broadly neutralizing antibody response. To me, I think you need a good neutralizing antibody response. I think you need T-cell responses as well. I don't think anyone thinks that there's going to be a neutralizing antibody response that neutralizes every variant of HCV to which someone might be exposed. So I think having a T-cell response to complement that is critical. I wouldn't make too much of the Okyris vaccine. It was a BK strain vaccine. Every circulating strain of HCV carries a number of escape mutations. That's how it circulates in human population. So I think there are numerous reasons that I don't want to extrapolate too broadly beyond the vaccine as prevention trial to say that a vaccine is not possible. I firmly believe it is possible to get protective immunity. And I think in phase 1 clinical trials, we could determine what our best in class T-cell and B-cell antigens are and bring that forward.
That was a lot more thoughtful than what I could have said, but I think just to synthesize the answer to your question that you just answered and the one that was asked before is, you know, what we learned with SARS-CoV-2 is that if you're in the middle of a public health crisis, that you get the smartest people you can to think about the problem, and then you act decisively, and you make sure resources aren't the limiting step. And then sometimes you're surprised by how well things work out. And I think that's what we learned with SARS-CoV-2. And I think we have a public health crisis right now. There's, instead of 6,000 people that are supposed to be acutely infected with Hep C in 2030, we're going to probably have more like 90 or 100,000. And we're not going to eliminate. So we need to respond and get the smartest people, think as hard as we can. And then, as Marion said, take a lot of attempts into phase 1 and see what we learn. I mean, that's my opinion.
I maybe want to add, because I think, Marion, your message is so important to be heard by more people, that it does not take as much as many people think to do a phase 1 trial. A lot of, I see this still so much, it's like, therapeutic HPV vaccine is the same. People seem to want to modify and test their vaccines for years in mice and whatnot of models they have. And it frustrates me immensely, because we all know that's not going to help us. We need to see what quality of immune response we get in humans, that we have a baseline, also that we can compare different vaccine approaches and see what kind of immune responses they make, so we can make a better decision.
Yeah, I do want to make that point that a phase 1, there are many vaccines that do not go beyond a phase 1. Many, many more that do not go beyond phase 1 than actually do. And you can learn so much from them, I think, that can inform. So I guess one thing would be, don't be afraid to fail. That doesn't mean that you can't learn from it.
Well, on that note, let me ask, what are the expectations and what are the metrics to advance beyond phase 1? To really people who have already made comments, and I mean, what is it that we're looking for, and in what population? Who would like to tackle that?
Well, I guess I'll have a go, Stephen, and you probably can predict my answer, and that is, if we can see in phase 1 pretty good cross-neutralizing antibodies to these panels from Justin and Thomas Peachman, that would be a thumbs up, right? And I'd like to see strong CD4s, obviously, but I'd like to see strong CD8 responses to highly conserved epitopes. And if, the more the better. If we've really primed CD8s to highly conserved epitopes amongst the genus, and we've got more than one, I think I would be extremely pleased and enthusiastic to take that into a CHIM or some other efficacy trial. Could I, if you don't mind, could I just come back a little bit? Because I would like to ask Andrea and Marion, your question on question 2, you're asking, should the phase 1 be in, I think you're asking, should it be in healthy volunteers or should it be in PWIDs? I'd really like to hear the comment from Marion and Andrea on that. The first phase 1 trial I'm talking about.
Well, I would comment there that a phase 1, the first phase 1, I don't think that PWIDs would be treated as different or special. I mean, I think that there's no reason to think that they're not necessarily a different population. They're not necessarily unhealthy. I'm not sure that that's the case. Andrea could comment on what how they'd be classed. But one thing would be, if you are going to do that, and I think that might be difficult for the initial phase 1, just in terms of planning a clinical trial in PWIDs may be more difficult than in healthy population, but also it might be that because they are a vulnerable population, it may be considered that initially it should go into a healthy population. So I think that question, was more, I was thinking, well, maybe the follow-up, you might look at your phase 1 in a healthy population, in a non-PWID population, but then maybe a follow-up would be in that group to see if the immune responses are similar. That's my thought. Maybe Andrea can add some comments.
Andrea, quick comment.
Quick comment. I agree with that. I think I would do head-to-head comparisons of vaccine strategies in healthy volunteers. Despite the extensive expertise of Kim Page, Margaret Heller, people around the world, Nagla Shukri with Julie Bruneau, and following PWIDs, it is more challenging than following other populations. So it's easier to do a head-to-head of strategies in phase 1. And ours is one of many studies now with HIV vaccine trials showing lower responses in PWID. So I think you get your best response in healthy volunteers not at risk for HCV and move to PWIDs next. But one could argue that you may get more information by going into PWIDs first. It's just harder to achieve good follow-up and other things than in healthy control populations.
I'd like to close up this discussion to thank everyone and all the participants, all the speakers, and also for this very lively discussion. I don't know whether there's enough time for the organizers to make some closing comments. John, Chris, Rajan?
I think John has had to leave, so I don't think he can make any comments. Chris may want to say something, but before that, I would just like to thank everybody and really reiterate what an important conference this was, how important and how excellent the science was. We've come to understand one thing more clearer than anything else, and that's non-scientific. If you might, and that is the final comments we had from Marion and Andrea, that we just need to get as many candidates into testing as possible, because keeping in mind that it doesn't have to be perfect, and we don't have to have all the boxes checked before we do small trials. And so thank you again to everybody. Chris, please close us out.
Thanks, Rajan. I've greatly enjoyed the discussion. It's kind of amazing how I think the thinking in the field has changed with Marion's comments about more rapid movement into humans, the concerns about what animal studies may or may not be telling us. And I think the future is exciting. And I'm really hoping and hopeful that Mike is right, that we'll have a vaccine by the end of this decade. So with that, I really do want to thank the Coalition for Global Hepatitis Elimination for bringing us together. It's been a long time since we've been together, and I've greatly enjoyed it. And I hope you have too. Thank you.
Goodbye, all.
Well, it's fun to see you all again.
Bye-bye.
Yeah, thanks a lot.
Even if by Zoom.
Bye, everybody.
Everybody.
Thank you.
Take care.
Take care.
Bye. Bye-bye.